Again, welcome everybody for the second day of the Neural Engineering Research Symposium at the University of Miami. Very excited to have all of you join us. I'm going to keep it very brief, but I really want to thank four people that led the research organization for this symposium: Dr. Zabhishek Prasad, Dr. Courtney Dumont, Dr. Andy Dykstra, and Dr. Patrick Ganser, who organized and reached out to the speakers, organized the student presentations. Really great stuff. And also, of course, we couldn't do this without people who behind the scenes make all of this happen. And so Erica Suazo, who has been wonderful helping with the Neural Engineering Institute, as well as with the symposium for the last 5 years. Really, thank you. And again, Ignacio, Iggy, without all the technical help you give us, we couldn't do all of this. So thank you for really leading all of this and helping us organize this. I think all of you have been wonderful helping with this symposium, making it a success for the last 5 years. And hopefully we're going to keep this going every single year before the neuroscience meeting, the Society for Neuroscience. I know that was a little bit of a setback this year with them canceling it in person at the last minute. But I hope that these kind of smaller regional discussions can really sort of spring ahead, like, collaborations as we move forward in the future between our institutions, between our departments. And so with that, we'll get started. We have a great program today, but before we have a brief remarks by the new Dean of Engineering, Dean Pratim Biswas. So Dean.
Welcome to the 5th Annual Neural Engineering Symposium at the University of Miami. As the Dean of the College of Engineering, I'm excited to welcome all of you, investigators, students, and postdocs from across neural engineering and neuroscience-related research fields. This symposium and recent establishment of the Institute of Neural Engineering at Miami is the result of cross-campus programs established jointly by the Colleges of Engineering, Arts and Sciences, and the Miller School of Medicine at the University of Miami for promoting collaborations and to promote the advancement of new technologies into our research and clinical programs. I'm really glad that this year's symposium will be based on a theme of neurotechnology and neuromodulation in trauma and disease. This theme is particularly relevant as researchers like you across multiple institutions and centers are involved in the development and testing of new technologies, novel therapeutics, rehabilitation approaches focusing on brain and spinal cord injury, neurological disorders, and overall brain health. I look forward to hearing from all the exciting keynote speakers and the presentations by trainees addressing emerging themes in neural engineering. I wish you a very enjoyable and informative conference that continues to lead to new collaborations and ideas for moving this exciting field forward.
So thank you, Dean Biswas. There's a great partnership between our schools at the University of Miami, and we'll continue to build on this theme in the future. With this, I'll turn it over to Drs. Dumont and Dykstra to lead today's session.
Perfect. Thank you, Dr. Rajguru. So we're actually going to move into the student presentations to kick off today's events. There's a lot of really exciting talks today, and our trainees are included in that. So the first talk, if you can pull that up, is by Raja Mailavapu, featuring the modulation of motor cortical neurons in common marmoset overlaps with macaque.
This presentation will focus on the task-related modulation of neurons in the primary motor cortex of the common marmoset. The primary motor cortex is a control site that's commonly used for brain-machine interfacing approaches due to its role underlying generation of voluntary forelimb movements. We assess the underlying firing properties of constituent neurons in the marmoset motor cortex and identify modulation in both a subset of neuronal population spiking activity and local field potentials. 3 adult male marmosets were trained on reaching tasks of 2 types, go-no-go reaching and multi-target reaching tasks. The go-no-go reaching paradigm consisted of cues representing the instruction to either move to a target or to maintain the original hand position. The multi-target reaching tasks were done with either 2 targets or 4 targets. A 16-channel electrode array was implanted into the primary motor cortex of the marmoset. Implant locations were determined by stereotaxic coordinates and confirmed with intrasurgical surface stimulation of the cortex. Spiking was isolated from broadband signals with online box sorting, and LFPs were isolated from broadband signals using 4th-order Butterworth filters. Log firing rates and regularities were calculated by fitting to the scale and shape factor of a gamma distribution. The overall firing rates and regularities of all neurons in the recorded population did not shift between recording sessions. A subset of the neuronal population showed modulation of firing rate during trials with movement in green versus no movement shown in red in the histograms. This was true for the mu and beta bands of the LFP power, which showed attenuation with movement onset, and the high gamma LFP power, which increased as shown in the spectrograms. Furthermore, there were subsets of neurons and channels of LFPs showing directional selectivity as well. It was also possible to decode trials with movement versus no movement and to decode rest versus movement phases across spiking activity and LFPs using linear support vector machines. This was above chance levels for both monkeys. The log firing rates and log firing regularities were within ranges reported for other mammalian species such as rats and macaques. Also, the presence of neurons tuned to movement onset and direction and the modulation of LFPs mirrored reports found in macaques. This suggests that the modulation of neuronal activity in the marmoset motor cortex is similar to that of macaques and provides further information regarding the use of this particular model species in future neural interfacing studies.
All right, so in terms of if you have any questions, feel free to add them into the chat. I do see that a number of our students that are presenting are here today. So perhaps they could either answer it in the chat, or if there's time at the end of the student presentations, we can allot some time for questions as well. With that, I'd like to move into the next student presentation by Kayla Meisinger. And she's going to present her work on the role of pyroptic cell death in cochlear implantation trauma.
My research focus is on exploring the involvement of pyroptosis in the pathophysiology of cochlear hair cell death induced by cochlear implantation trauma. Electric acoustic stimulation cochlear implant devices have been designed to benefit patients with preoperatively high-frequency hearing loss but significant residual low-frequency hearing. However, a significant risk of loss of residual acoustic hearing post-cochlear implantation remains. Past research has shown that cochlear sensorineural cell death plays a critical role in the pathophysiology of cochlear implant injury, and more recently, pyroptosis has been identified as a form of programmed cell death related to this. During pyroptotic cell death, an inflammasome sensor detects stress signals and activates caspase-1 through the ASC adapter. Then activated caspase-1 leads to cleavage of gasdermin D, maturation and secretion of the inflammatory cytokine interleukin 18 and 1 beta, and ultimately leads to pyroptotic cell death. Despite this new knowledge regarding cell death in cochlear implantation trauma, a detailed understanding of how pyroptosis is involved in cochlear implant injuries remain elusive. So, in the study with the use of adult female brown Norway rats, we performed Western blots, RT-PCR, and immunohistochemical evaluation to quantify the role of pyroptosis cell death in a preclinical rodent model of cochlear electrode insertion injury. Gene and protein expression levels of selected factors and immunohistochemistry labeling of specific markers were quantified in cochlear implant injured and untreated contralateral cochlea at 6 and 24 hours post-electrode insertion. As shown in Figure 1, Western blot data, and Figure 2, RT-PCR analysis, cochlear implant increases the protein and gene expression level of key pyroptosis markers, including NLRP3, ASC, caspase-1, gasdermin D, IL-18, and IL-1 beta, both at 6 and 24 hours post-electrode insertion. We then also quantified gasdermin D and caspase-1 activity in the inner and outer hair cells 24 hours post-cochlear implantation. As reported in Figure 3, the role of gasdermin D and caspase-1 in cochlear sensorineural degeneration is not limited to just the cochlear hair cells. So these preliminary results suggest a critical role in pyroptosis cell death and cochlear implant trauma. Future experiments will assess pyroptosis key regulatory steps of specific cellular population in the inner ear and will provide new insights on how pyroptotic cell death regulation may be used as potential novel neuroprotective treatments to protect and preserve residual hearing following cochlear implant insertion. Thank you.
Awesome. Thank you, Kayla. So the next talk we have is by Andrew Cicerello, and he's going to present his work on synthetic ligand receptor binding for targeted delivery to the injured spinal cord. And again, if you have questions, please feel free to put them in the chat.
Hello, my name is Andrew Cicerello, and I'm a graduate student in the Department of Biomedical Engineering at the University of Miami. And today I'll be presenting my poster entitled Synthetic Ligand Receptor Binding for Targeted Delivery to the Injured Spinal Cord. So the overall goal of this project was to develop an on-demand drug delivery system to specifically target an injured spinal cord. In tissue engineering and drug delivery, targeted delivery is not a novel concept. However, a lot of targeting strategies rely on naturally occurring ligands expressed on the cell or tissue level. Now, this is problematic, especially in spinal cord injury, as naturally occurring ligands or receptors change over time and are susceptible to remodeling or are ubiquitous throughout the entire nervous system. So for this system, we decided to try to shift away from a naturally occurring system and develop a synthetic system. So we engineered an implantable beacon that expresses biotin that goes into our injury site and acts as a targeting reservoir for nanoparticle trafficking. The nanoparticles we developed express the complementary streptavidin to bind to our implantable beacon. So we're taking advantage of the biotin streptavidin complex, one of the highest known affinity interactions in nature. And so once we have our implanted beacon in our injury site, the idea would be to inject our streptavidin nanoparticles intravenously, and they would traffic and bind at our implantable beacon. So starting off in vitro, we first demonstrated that our NPs did indeed bind to our engineered beacon, and that they actually were retained over time and did not just get washed away once they bound to our beacon. Then moving into our in vivo studies, we demonstrated a significant increase in our streptavidin functionalized nanoparticles binding at our implanted beacon compared to our control conditions. And furthermore, we saw that with our implanted beacon, our nanoparticles actually were retained at our injury site. So with our increased targeting and our retention, we have demonstrated an improved ability to traffic our nanoparticles to our implant, and thus would be able to better treat spinal cord injuries. I'd be happy to answer any questions. Thank you.
Perfect. All right, thank you, Iggy, for replaying that, and thank you, Andrew, for submitting your work. Surprisingly, we're still moving right along on time, and I ask that you guys continue to post any of your questions in the chat. The next talk is by Liana Roman, and her work is going to detail measurements of mechanical steady state accommodation fluctuations using optical coherence tomography.
Hello everyone, I am Liana Rahman. I am a second year PhD student in biomedical engineering at the University of Miami, and I'm doing my research work at Bascom Palmer Eye Institute. Today I'm going to present my abstract called Measurements of Mechanical Steady-State Accommodation Fluctuations Using Optical Coherence Tomography. So in our everyday life, our eyes are constantly changing focus to maintain a clear vision. An image out of focus is the stimulus that triggers the accommodation reflex that will allow us to see clearly. This stimulus activates three efferent reflexes controlled by the parasympathetic nervous system, which are the pupil constriction, the lens accommodation, and the convergence. During lens accommodation, the parasympathetic fibers will give the signal to the ciliary muscle to contract so that the tension on the zonular fiber can be released to let the lens take the needed shape to achieve clear vision. However, as we grow older, the lens becomes stiffer and thicker and is not able to take the appropriate shape anymore for a good sight. This condition is known as presbyopia. Therefore, to understand the causes of presbyopia, we study the mechanical microfluctuations of the intraocular distances at a high speed using an extended depth OCT system. Our purpose was to see if we can detect the low frequency and high frequency component of the mechanical fluctuations of accommodation, characteristic of optical microfluctuations. Also, we examined if those microfluctuations were correlated with the heart rate, as it was suggested by some prior studies. To do that, we performed two experiments. For the first one, we recorded the fluctuations of the anterior chamber depth and the lens thickness of the right eye of two young adults for an accommodative demand of 0 diopter and then 4 diopters. At the same time, we recorded their heart rate with a pulse oximeter. The imaging session lasted 10 seconds. Then to measure the anterior chamber depth and the lens thickness, we use a custom-made semi-automated MATLAB application to detect the boundaries of the intraocular distances. Afterwards, from the time domain variation, we calculated the power spectra of the anterior chamber depth, the lens thickness, and the heart rate variation. The second experiment was kind of similar to the first one, but this time we recorded the full length of the eye of one young subject and one presbyopic subject with the same system. The difference here is that we capture 4 mm central zone with 320 lines per frame to facilitate the segmentation of the boundary of the retinal boundary, and this with an acquisition speed of 10 Hz. Similarly, to the first experiment, a pulse oximeter was clamped to the finger of the subject to keep track of their heart rate. So our results confirm the presence of the low frequency and high frequency component producing the lens thickness variation. We also noticed an increase in the amplitude of the fluctuations when increasing the accommodation stimulus. However, we couldn't find any correlation between the heart rate and all the intraocular distances. As you can see here, the red dashed line corresponds to the heart rate frequency, but there is no peak in the power spectrum of the lens thickness or anterior chamber depth corresponding to this heart rate frequency. Instead, in the power spectra of the axial eye length, we found a consistent peak around 2.5 Hz for the young subject that can be caused by eye movements such as saccades, which are controlled by the neural system. So as a conclusion, we demonstrated that we could use OCT to quantify the microfluctuations of the ocular biometry at steady state accommodation. Now a larger study is underway to confirm these findings and assess the relationship with the lens mechanical properties and their changing with age. Thank you for your attention.
Perfect. All right, thank you. So that's concluding our 4 student talks today. There was actually quite a few more student talks that we had overall, and all of our student talks were considered for awards, so we'll be announcing those later in the schedule today. But I just want to congratulate all the students for their really exceptional work and all of their efforts that they've put forth and for sharing it with us today. So it looks like we have about 4 minutes before we switch gears a little bit. So I wasn't sure if any of you have any questions that you'd like to bring or to ask the students that we've seen today. I know there's been quite a few questions for Kayla in the chat, but are there any other questions that you all might have for the student presenters today? Okay, if there's no other questions, then we'll just take a quick 3-minute break before I pass things off to Dr. Dykstra, who's going to introduce our featured speakers for the day.
Okay, we're back. Hi, everyone. It's my pleasure today to introduce the 3 symposium speakers for our session on neurotechnology and neuromodulation in injury and disease for this morning. Our first speaker comes from the Department of Chemistry here at the University of Miami, and Caitlin Meyer will be speaking about elucidating the role of metal-protein interactions in Huntington's disease. Take it away, Caitlin.
All right, so good morning, everyone. I first want to thank the organizers of the symposium for giving me the opportunity to share some of the work that my group is doing here at the university. And first, before I begin, is everyone able to see the slides and hear me all right?
We can see you and hear you perfectly.
Wonderful. All right. So as Andrew was saying, I am an assistant professor in the Department of Chemistry here at the University of Miami. And the work I'm going to be telling you about today is related to my group's interest in defining the role of metals, and in particular metal-protein interactions as they relate to neurodegenerative disease. Now, the work I'm going to be telling you about is really done in collaboration with the lab of Dr. Grace Zai from the Department of Cellular and Molecular Pharmacology at the Miller School, as well as Dr. Rajiv Prabhakar, who is also here in the chemistry department on the Coral Gables campus. So before I delve into the nitty-gritty of our research, I wanted to give you an overview of Huntington's disease. So Huntington's disease is an autosomal dominant genetic disorder that has that actually impacts about 30,000 people nationwide, with the potential to impact a great number more in the coming years. Its most characteristic feature rather is a series of CAG trinucleotide repeats that sit on the Huntington protein that is linked with the disease. Now, when you have extended sequences of glutamine residues, these form a consecutive chain, and the chain length here has been linked to oligomerization, as well as disease severity, and also is used to predict age of onset of the disorder. Now, we all have Huntington protein in our bodies, but for most of us, we have the normal form of the protein, which has fewer than 36 glutamine residues. We call this the non-pathogenic form. And then for those persons who present with Huntington's disease, they are known to have the abnormal or mutant form of the protein, which has greater than 36 glutamine residues. Now, when patients present with Huntington's disease, general symptoms include slurred speech, motor function impairment, and neuronal degeneration. Additional markers include mitochondrial dysfunction and axonal transport impairment. Now, in addition to those markers, one of what I think is arguably a more overlooked feature of Huntington's disease, and in particular research related to it, is the involvement of metals. So iron and copper especially are shown to be significantly elevated in Huntington's disease. Iron also reported to be significantly elevated in diseases such as Parkinson's, Alzheimer's, and Friedreich's ataxia. For today's talk, I'm going to be focusing on the role of copper. And this really caught my interest because there have been a number of reports that quotes an increase of 26% in copper levels in the striatum and as much as 51% in the cortex of transgenic mice models. This has also been observed, that being an elevated level of copper in postmortem brain slices of HD patients. Now, the really critical piece of information here that's missing is the exact role of copper in promoting pathogenesis, that being whether it's a direct effect via direct interaction of the metal with the protein, or indirect, perhaps through modulating mitochondrial dysfunction and generation of reactive oxygen species. Now, the real significance of this, and I think what really motivates my interest in the work, is that many of the common therapeutic approaches that are aimed at treating the symptoms or alleviating the symptoms associated with Huntington's disease rely on metal chelation therapies. Now, this is a bit worrisome, in my opinion, because if we don't quite know what the role of that metal is, how can we selectively target it? So what I'm going to show you now is a really oversimplification of how I view chelation therapies as functioning. So imagine we have in our bodies, in our brains, Huntington protein floating around, consider it this purple sphere, and then you also have copper, consider it these orange spheres. If the metal is interacting directly with the protein, you can have a metal protein complex. You can also have labile pools of these metals floating around. Now you administer a chelation agent. As I said, this has become one of the prominent treatment options for treatment of HD-associated symptoms. And the most common of these are trientine, penicillamine, and cleoquinol. These are often administered either orally or intravenously. And so these are being administered into the total system. So the chelators here are these green hexagons. They're really just chemical compounds that are designed to target a specific metal in a particular oxidation state. And so they can come in, bind to either the metal in the free labile metal pool, or it can strip the metal from the Huntington protein in this case. When it does this, it renders the metal inert and allows for the metal to be safely removed from the body without reacting further. But we know, that's a very simple example. In our bodies and our brains, we have many other proteins and enzymes that also require metals for proper folding and function. And so if we consider now a broader pool of proteins, consider these purple or pink squares, they may also require the same metal in the same oxidation state for proper function. Now you administer a chelator that's targeting those copper ions, and that can come in, and if there's no other selectivity method or mechanism in place, the chelator can strip the metal from the labile metal pool, from the Huntington protein, which in this case would be our desired target, or from the other proteins that actually require it for proper function. And so this can be very closely linked with deleterious and undesirable side effects. So you may be treating some of the symptoms associated with HD, but you may also be getting some other very deleterious effects. And so this really underscores the importance in our work to understand the interaction of both wild type and mutant forms of the Huntington protein with metals. In this case, we're looking specifically at copper, and to explore ways that we can really guide the design of more selective chelation agents. So here I'm highlighting some of the questions that my group is really interested in. So first, is copper-2, in this case, actually involved in pathological oligomerization of the protein? So as that poly-Q tail increases and we see this uptick in oligomerization, is this triggered by copper? And if so, is the copper directly bound to the protein? And the second case, is this kind of an indirect effect? Indirect being through generation of reactive oxygen species in the presence of copper. And if so, what are these reactive oxygen species and what concentration are they accumulating? And I think this is also something that was kind of touched on, albeit in a different system, in one of those posters from yesterday. And so this is something we are also very much interested in. And finally, when we consider the chelators, are these preferentially targeting a specific interaction, or can we consider them as targeting the general metal pool? And if it's targeting a general metal pool, how can we make them more specific? So to tackle this really complex problem, it requires a multifaceted, multi-pronged approach. So my group is expert in a variety of biophysical methods, mostly spectroscopic techniques that span a wide range of energies. So we use techniques ranging from microwave radiation up to gamma radiation. We're getting very precise and in some cases metal-centric insight into what's going on in our in vitro protein systems. We couple this to in vivo studies that are performed in HD transgenic Drosophilus systems that are well established by our collaborators in the Xi group on the medical campus. And what we can do there is couple our molecular level insight from our spectroscopy to these in vivo studies, which elucidate via staining of brain dissections, accumulation of species such as copper, reactive oxygen species, Huntington protein aggregates, and so on. And they also have the capability to look at behavioral assays of these Drosophila systems in order to see how copper is impacting disease pathogenesis. Finally, we also look at in silico Huntington protein models, developing kind of a complete picture where there really wasn't one of our in vitro system to see how metal binding could take place, what the site looks like, and what's around the site to enable us to develop a more selective or targeted chelation agent. So to get started, I think it's most helpful to begin with our in silico model. So this is a computational model that we prepared in collaboration with the lab of Professor Rajiv Prabhakar and his student Leo. Both were very instrumental in training my graduate student Deepa and I in molecular dynamics approaches. What you see here is the complete in silico model of our particular in vitro system. What's important to note here is that this is only a fraction of the protein itself. The protein is massive. It's about 350 kilodaltons. And a lot of the studies that have been published on Huntington actually only focus on the first 7 to 17 amino acids at the N terminus. And so somewhere between this purple and orange region. And many of those studies actually didn't even consider copper as a variable. What you'll also notice is that the color coding here represents stitching together of multiple crystal structures. So the presence of these glutamine residues make this protein very flexible and very difficult to resolve by traditional X-ray techniques. And so what we've done here is use molecular dynamics simulations to generate, to the best of our knowledge, the very first complete model of our N171 in vitro system. So 171 referring to the number of amino acids in the Huntington protein, along with some larger flanking sequences coming from crystal structures. Now this is significant because not only is this kind of the first complete model of this missing fragment, but it also is the region that contains many of our putative metal binding residues, things like methionine, histidine, cysteine, and so on. So with that in silico model in mind, what we then did is we looked at our in vitro system. So again, we're looking at 171 amino acids of this Huntington protein, and we have a number of constructs available in our lab, but today I'm only going to focus on our normal or non-pathogenic form. This has 17 glutamines in sequence. And the significance of this work is that we're really pushing our concentration limits of this protein. Now we're at about 100 times what's been previously reported. And this is really necessary for a lot of our spectroscopic investigation that's trying to probe the metal site specifically. What I'm showing you here is that we're able to obtain very pure fractions of our Huntington protein in the absence of copper. And then interestingly, and also consistent with some of the early reports on the Huntington protein by Jonathan Fox and others, we were able to show that in as little as 1 hour of incubation at either 4 or 37 degrees Celsius, we see conversion of some of our monomeric protein to the dimer form. And this really told us that copper addition to our monomer protein is resulting in dimerization of that protein, even in the non-pathogenic form. And so this was really promising in that copper is inducing some sort of a change. Now the question is copper inducing a large scale structural change as what's been proposed previously? And so in order to answer that question, what we did is we turned to our biophysical toolbox and we probed our system using circular dichroism spectroscopy. So for those of you who might be less familiar, CD uses the very same principles as UV-Vis absorption spectroscopy, except we are utilizing circularly polarized light in this case. So it has both left and right circularly polarized components. And when we record the spectrum of our sample, which is inherently asymmetric, it's a nice protein sample, we can record this across a wide variety of energy ranges. And by changing the energy range, we get different types of information. In this case, I'm showing you the far UV energy region, which is going to give us a very nice feedback or readout on the type of secondary structure that we see in the protein. So here, this is a pure alpha helix, pure beta sheet, or pure random coil. Now, the important thing to note here is that when we recorded our circular dichroism spectrum of our apoprotein shown in blue, this this result told us that the apoprotein is predominantly composed of alpha helical and random coil content. When we then keep protein concentration fixed, but add copper to kind of correlate with our SDS page study that I just showed you, we see a significant reduction in both alpha helical and random coil intensity, and we see conversion to beta sheet-rich structure, which tells us that this is consistent with oligomerization or a trend towards aggregation of the protein. So this is indeed saying that copper is playing a role. We can use this, and we see this in our in vitro model, and this is really validating our system to move forward with the metal-specific interaction studies. So moving forward, we're shifting our focus to the UV visible range to get information about the metal-protein interaction specifically. Now, that's all of our molecular level insight in terms of what's going on in the protein at a larger scale. Now, to couple this to the in vivo Drosophila system that the Xi Lab has very well established, we worked very closely with Amanda, who is a graduate student in Grace's group, and Yi, who is a postdoc in Grace's group. And together we were able to work out a very robust and reproducible feeding strategy for enriching the diets of Drosophila systems with copper. So here we either had a control group or groups that were fed 250 micromolar copper or 500 micromolar copper. And this again was all done via dietary enrichment. What I also would like to note here is that the Zilab has two HD models that we've used. And so they predominantly differ in terms of this particular system being a GMR-driven system that has red fluorescent protein tagged huntingtin. And this is expressing predominantly in the eye. So we get a lot of feedback on eye morphology and photoreceptors. We have both a non-pathogenic form that only has 15 glutamines or a pathogenic form that has a much longer chain of 138. And what I'm showing here is the control group. And as we move from left to right, we're increasing the copper concentration in diet and staining either for the RFP huntingtin protein or using an amyloidogenic aggregate stain THS to see accumulation of huntingtin aggregate. And what we can see is that even in the pathogenic form of, or non-pathogenic form of the HD model, there is a slight increase in amyloidogenic aggregates that accumulate. And this is perhaps consistent with our non-pathogenic in vitro system. The more striking result, I think, is when we shift to the results from the Q138 or pathogenic HD system. And again, changing copper concentration and looking at the age of the fly. So either from 5, 10, or 15 days after eclosion. And the plot down at the bottom here, I think summarizes this quite nicely, where you can see that THS positive aggregates per lamina are increasing in both an age-dependent manner, as well as a copper dose-dependent manner. And this is, I think, perhaps most obvious in the highest copper dose group. We're also able to look at some other structural features in the Drosophila eyes using the same model. Here I'm showing the non-pathogenic again with 15 glutamines, increasing copper, and staining either for huntingtin protein, for BRP, which is going to report on the structural integrity of the synapse, or cleaved caspase 3, which is going to report on apoptosis. And what you can see here is in the non-pathogenic form, perhaps there's not as drastic or obvious an increase in huntingtin protein accumulation. But when I show you now the pathogenic system, you can see there's a marked difference. So not only are we seeing the effect of the Q138 contributing to HD progression, but we are also seeing an accumulation of huntingtin protein accumulating in the lamina, as well as a reduction or degeneration rather of the synapse. And we're also seeing an increase in cleaved caspase 3 accumulation that is kind of co-localizing with that huntingtin protein. So this is telling us that copper is inducing apoptosis and synapse degeneration in a copper dose dependent manner. So at this point, we had really started to lay the groundwork for understanding the role of copper in huntingtin protein accumulation, as well as oligomerization and potential aggregation. And we wanted to begin addressing this question of reactive oxygen species accumulation. And so in this particular case, again, I'm showing you from left to right, increasing copper concentration. But this system is actually utilizing a second Drosophila HD transgenic model that it was also heavily used by the Zai group. In this case, this does not have the red fluorescent protein tagged huntingtin, and the non-pathogenic form of the protein has 20 glutamines, whereas the pathogenic has 93. So again, a much longer chain compared to the non-pathogenic. And here what we're doing is we're staining for reactive oxygen species accumulation using DHE. Now what you can see, and what I want to draw your attention to, is this bottom right panel, where at the highest copper dosing in the pathogenic model, we see a significant accumulation in the DHE stain intensity, which is standing for the superoxide here. And we see this in a copper dose dependent manner as well. To correlate this with our in vitro system and to our molecular level study, we then decided to take our apoprotein, again, trying to look for the impact of reactive oxygen species generation and metal interaction with protein. And we decided to study this in the absence and presence of copper using a tool called electron paramagnetic resonance spectroscopy. So you don't have to be an expert or to have even heard of EPR before. The general idea is that we have a protein, in this case, our Huntington protein. It should have an unpaired electron in the system. We put that sample between the poles of a magnet and we irradiate that sample with microwave radiation. In this case, very fortunate for us, copper-2, which is the salt we're adding to our Huntington protein, has one unpaired electron, and the spectra we get provide very rich information about spin state, geometry, and metal and ligand coordination environment. What I'm showing you here is the EPR spectrum of the copper salt that we're adding to our protein in black, and our apoprotein that says no metal added, and it's actually treated with EDTA to remove any metals that might have been present during expression. What you can see is that we have a very clear signal for the copper salt, and the apoprotein gives no signal at all, as we would expect. When we go ahead and we add copper to our apoprotein in a 1:2 ratio, we obtain this blue spectrum. And we can go ahead and increase this ratio as well to 1:8. And what you see is that we see a clear shift in the spectra from black to red and blue. And this is also observed in some of the fine structure. And what this tells us is that the copper species that we're seeing in our actual EPR data of this in vitro model are in very different coordination environments than the salt that we added. And so this tells us that the copper must have reacted in solution in the presence of the apoprotein. And it's important to note that this happens only in the presence of the apoprotein. And so this tells us that this is very likely occurring as a result of reaction with ROS that are generated in the solution. And so one way that we're testing this now is to include a radical trap, something similar to what's been used extensively in the literature, such as DMPO. The beauty of the radical trap is that it will go after the radical species, which are typically really short-lived, very reactive. And when it does that, it will actually form a very stable radical adduct that has a very characteristic EPR spectrum. So it makes it very easy for us to not only define the identity of that ROS, but also to quantitate how much is there. So we can say what it is and how much is actually accumulating. So in summary, today what I hope I've shown you is that using this combined approach, we are able to prove that in our in vitro model, copper-2 is actually able to induce dimerization of the monomeric protein. And we can confirm that this dimerization and subsequent aggregation does result in beta sheet rich character as judged by CD. We can also show using HD Drosophila systems that there is an increase in THS positive aggregates that occurs as both an age dependent and a copper dose dependent manner. Finally, the pathogenic HD Drosophila model shows copper-dependent effect, not only on apoptosis, but also on synapse degeneration. And we also see superoxide accumulation, really significant, I would say, superoxide accumulation in the highest copper-enriched Drosophila group. Connecting all of this now back with our in vitro system, we also see this appearance of a new EPR active species, which indicates to us the likely generation of ROS, that really will be the subject of our next set of experiments to really define and again quantitate how much of those species we're generating. Finally, I'd like to thank my collaborators and my mentoring team, Dr. Grace Tsai, as well as her graduate student Amanda and postdoc Yi. I'd also like to thank my secondary mentor, Professor Rajiv Prabhakar, and his graduate student Leo, who again were very instrumental in helping my student and I become trained in molecular dynamics simulation so we can carry out future studies. My lab, which was started in 2019 here, and so we're still a pretty small group, But I'd also especially like to highlight Deepa, who contributed the in vitro and in vitro work I showed today, as well as worked with Leo to develop the in silico models. I'd also like to thank the CTSI for funding support through my KL2 mechanism, as well as the University of Miami Department of Chemistry and the Frost Institute for Chemistry and Molecular Science, who really provided the initial funds to get us started on this project. Finally, I'd like to thank you for your attention. I know this was a bit of a different talk than we've seen, focusing a little bit more on how we can improve existing chelation therapies to treat neurodegenerative diseases, but hopefully there was something that you were able to latch onto and resonated with you. So with that, I'd like to, again, thank you, and I'd be happy to take any questions you might have.
Thanks, Dr. Meyer. Very nice talk. We do have a couple minutes for questions if anybody wants to ask live or post in the chat, and I can read them as well.
I have a question, Caitlin. Can you hear me?
Yes, I can hear you.
Oh, it's a great talk. I enjoyed very much.
Thank you.
I have many questions. I know we don't have time for it, but I want to ask only two questions.
That's great, yeah.
Two questions. So what is the cause for increased copper in the brain to begin with? And second one is, have you seen enhanced accumulation or complete dimerization of Huntington upon copper treatment? You saw partial accumulation. Did you see complete? Those are my two questions. Thank you.
Yeah, no, those are great questions. So to answer your first question, I'm not sure that there's a clear explanation as to what the exact cause for the elevated metal levels are in Huntington's. I think one hypothesis is that when patients undergo this neuronal degeneration, perhaps that kind of kickstarts a secondary process that perhaps can also impact copper transporters or efflux machinery. But as far as I'm aware, I don't know if there's been a conclusive ruling on what the exact cause of elevated copper is. Mm-hmm. In regard to complete conversion from monomer to dimer, so the slide I showed you was collected at a ratio of 1:2 protein to copper. So we could continue to push that concentration, but I'm not sure if we would get complete conversion to the dimer form. I suspect, based on what I've seen in the literature, that we'd likely see conversion to higher order oligomers before we'd see complete conversion to a dimer structure. Okay. That's my hypothesis.
Okay, thank you.
Okay, so thanks, Dr. Meyer, again. If anybody has any further questions, they can feel free to post them in the chat. So moving on to our next speaker. Our next speaker is Shridi Subramanian, and who comes to us from the Department of Neuroscience at Scripps Research. So, he'll be talking to us today about a radical new way of neuronal communication in the striatum. So take it away.
Thank you. I'm going to share my screen. Can you all see me and hear me?
Yes, we can hear you and see the presentation perfectly. You're ready to start.
Thank you very much. And I want to thank everyone, organizers, and Courtney for nice, kind emails to get me into the program. And it's kind of a little bit nostalgic now, because 20 years ago, I met Dr. Dalton Dietrich in his office. He probably might have forgotten me. So I had a postdoc interview in Miami, University of Miami, and I met him in his office. I think we had some food together. That was almost 20 years ago. So I'm kind of, when I saw this, my memories came back. Thank you, Dalton, for coffee or something you gave to me on that day. Appreciate it. So I want to really take this opportunity to discuss some of the new findings that we made recently, which is surprising finding that we also did not look for it. I want to start with my personal experience with my family. Like this is my first child. When we were playing with balls, every parents play with balls with their kids. And then when I throw these two balls away, for example, ball 1 and ball 2, I asked her to go and pick it up. Like she always went far away to the far away ball 2, and picked up that first. And then when she coming back, she picked up the ball 1. And I wondered about this phenomena. Okay, why does she always pick up the ball 2 first and ball 1 later? Still, I don't have any answer for it. Then the thing is, I wanted to do that experiment with my second daughter. So we did a similar experiment. Like I threw the ball, 2 balls, and she went in. I asked her to go and pick up this ball, and she always picked up the first ball first and the second ball later. Sometimes she never even walked towards the ball 2. I wondered about this phenomena, this complexity, like how to explain this process. I think that's how complex we are. We know that because our brain is situated in a way that it does so many things, we still do not understand it clearly. We know that there are many different regions, many different cells, but how they coordinate to come up with a particular behavior, we still do not understand. Unfortunately, this beautiful brain is affected by many different diseases, as we have been hearing from past 2 days, that many different diseases affects the brain. And unfortunately, we do not have any one clear therapy for any of these diseases. In this century, the beginning of this century, we have been working towards this. I hope one day human civilization would find this cure for this. So our contribution in this area is focused on striatum, as Kathleen beautifully described in her talk. So we were interested in the region, the striatum, which plays a critical role on cognitive, psychiatric, and motor behavior, which is in the deep of the brain. There are 2 kinds of neurons here. Dopaminergic neuron 1 type is called medium spine neuron, and dopaminergic neuron 2, medium spine neuron. So they have D1 MSN, D2 MSN. They are the major population, and they regulate various processes which ultimately determines our behavior. So striatum is affected by many diseases, which we just heard. Huntington disease predominantly affects striatum. And the Parkinson disease, we all know. Then autism spectrum disorder and schizophrenia. These are some of the major diseases which affect the striatum. So we were being interested in understanding some of the molecular aspects of these diseases. So we all know that the way that neurons connect to each other, they talk to each other. They have to talk to each other to perform any functions. We very well aware of the presence of electrical synapses and chemical synapses, and also we are now venturing into the area of exosomes. This kind of communication has been shown to be critical for the neuronal functions. What we found is that there is a new kind of communication. It's kind of via cellular protrusion. Long-range communication can also be possible in the striatum. So what are the evidence for that? Which is I'm going to talk to you today. So here is what you're seeing in the green is a striatal-enriched protein forming this long tube-like connection, trying to interact with the neighboring cell. This kind of long-range connections called tunneling nanotubes, which is getting more popular nowadays. And we discovered that the striatal-enriched protein engineers these tunnel-like structure, and then try to interact with neighboring cell. So this protein that makes these tubes is called RAS homolog, enriched in striatum. So as you see, this protein is highly abundant in the striatum of the brain, and also it's in the human. Its similar expression has been found in human brain. REST is expressed in a developmental manner. So it's postnatally induced after P6 and P10 continue to express highly in the striatum and also in the cortical and to some extent in the hippocampal area. So in adult brain, it is highly enriched in the striatum. If you look at the primary structure of REST, it is a small GTPase family of protein, but it also contains the SUMO E3-like domain, which is at the C-terminal end. So what are the known functions for REST in the biology and disease? There are many different important functions have been described. In our lab, we just showed that REST is critical for striatal degeneration in Huntington disease. And we showed that REST is also important for levodopa-induced dyskinesia. And also we showed that REST acts like an inhibitor of motor stimulation. It's like a brake in the striatum. And several other studies showed that there's a somatic mutation found in the autistic-like twins, and also there's the SNPs identified in a schizophrenia patient. More recently, REST is implicated in tau toxicity. And interestingly, the aberrant REST distribution is shown to be hallmark of tauopathies. So these are some of the known functions of the REST. So what we found serendipitously is that REST can make these tunneling nanotubes. Here is an example of showing this. You can see this beautiful long tube-like structures formed by REST, and they're trying to interact with neighboring cell. And these structures resemble cytonemes described in Drosophila, or they resemble tunneling nanotube described in PC12 cells. When you look at the alpha-structure analysis, and you can see there is a clear seamless transition of this tube from one cell to another. And at any given time, you can see a REST can make many tubes between the cell. So those are the striatal neuronal cell lines. So we looked at whether the primary medium spiny neuron that I described at the beginning, that the medium spiny neuron form can make TNTs. So here is an example. We cultured a medium spiny neuron in the primary neuron. You can see that the EGFP positive neurons these are all D2 neurons. This is made with the Cree. You can switch on the EGFP using Cree expression. You can see nice, beautiful expression here. But if you do the same thing with the Creon EGFP, and you can start seeing this long protrusion coming from the medium spine neuron. You can see these protrusions typically have the vesicle-like structure at the end of the tube. So REST can make tunneling nanotubes in most types of cell types we tested. Striatal neuronal cell, primary neurons, it can make these kind of tubes efficiently. That suggests that the REST utilizes the existing mechanisms in the cell to promote these long-range connections. Okay, if it is so obvious, why are we not able to see every time we do? The problem with most of the studies we do in our day-to-day lab work is that we try to fix the cells for imaging. So if we do that for tunneling nanotubes, for example, here is a primary medium spiny neuron, D2 neuron. You can see REST made many tubes here. But when you add a PFA, which is a commonly used fixator, you can see this neuron, these protrusions start destroyed immediately. It's also the same with the striatal neuronal cell type. So you can clearly see these protrusions disintegrate as soon as you put PFA. So most of the work that we do with in cell culture, if you add PFA, you don't see these structures because they're already gone. So that means you need to do live cell imaging to capture these protrusions. I wanted to show you here, okay, what is the use of this? What is it actually doing? So this video shows you that when there is these protrusions can interact with a neighboring cell, and the REST can move from one cell to another. here is a co-culture experiment. You can see the beautiful tubes interacting with the neighboring cell, and you see the vesicular punctures are moving along the acceptor cell, and you can see after some time these vesicular punctures start entering into the neighboring cell. So there is a clear delivery of rest cargo from one cell to another via these protrusions. What else these protrusions can be transported? So here is an example that these protrusions can transport endosomes. It is a connection protrusion made by RES, and you can see it's carrying this vesicle, endosome vesicle. They kind of, you can see they're following one after another. You can see that moves along, and they're delivered to the neighboring cell. What else can it be transported? Here is an example. I'm showing you that these membrane protrusions can also transport histamine receptor, H3R. These are the receptors being transported here. What else? So we also found that these protrusions can transport caveolin, for example. So here is the caveolin being carried through the protrusions, and they can interact with the neighboring cell. I mean, delivered to the neighboring cell. Okay, all these are cell culture, primary neuron. The question is, does REST can move between neurons in the brain, in vivo situations? So to address that, we had to again create novel tools to make this happen. So to test this hypothesis, so what we did is we had this we use again a Creon reporter. Basically, you switch on your protein of expression in any particular cell type in a Cre-dependent manner, as I mentioned previously. Here, we took a D1R Cre mice, the transgenic mice, which has Cre in a D1 MSN. Then we express RFP reporter or RFP REST reporter, your Creon. Then you just look at what happens. So here is an example showing you that if you express RFP in D1R Cre mice in the striatum, the RFP is just there in striatum as expected, because that's where the Cre is present. And we also looked at whether this RFP alone moves anywhere, but it doesn't move. But if you look at what happened with RFP-RES, RFP-RES we expressed in the striatum in D1R Cre mice, and we could clearly see the RFP-RES in the cortex. Okay, these are the layer 4 and 5. You can clearly see the RES started moving into the cortex, which is quantified here. Then we looked at the same phenomena in other mice, which is RGS9 Cre mice. Again, Cre is expressed in median spiny neurons. You can see RFP in the striatum is expressed, but not in the cortex. But RFP-RES expressed in the striatum start moving into the cortex. A clear deposition of movement of RES from one region of the brain to another, which is also shown here. You can see the quantification, absolute transport of RFP-RES to this cortex. The next question was, does RES move within the neuron, between the neurons in the striatum? For that, again, we need to create a new model, new mice. So here we crossed a D1R Cre mice. You can switch on, because we can switch on our protein. We crossed with the reporter D2R EGFP. The idea is if RES is expressed here, and see whether it's present in the D2R neuron. And what we found is, here is three different mice, the results are consistent. So if you express RES in the D1 neuron, here is D1 neuron, they start moving into D2 neuron. So there is a transfer, I would say collateral transfer of RES from D1 neuron to D2 neuron in the striatum. Because we think that the RES is moving using this protrusion, which I briefly described. So we wanted to evaluate whether these protrusions can be observed in a 3D environment. So far, we do not have sophistication to visualize these protrusions in vivo, in real mice. So what we did is we took organotypic slices. We made a slice from D1R Cree and D1R DT tomato slices. You can see there's a clear distribution of D2R and D1 neuron here. Then we looked at in some of the cells the appearance of protrusion, whether the rest is moving into D1R neuron. You can clearly see that 0 point we don't see protrusions, and at 10 or 20 minutes of image you can see the protrusions start coming up and they're interacting with the neighboring cell. The 3D intensity of this images, you can clearly the protrusions start coming here, obviously 20 minutes and 30 minutes, and that is co-localized with a D1R MSM. So this suggests that the rest can indeed induce this protrusion formation in the 3D environment as well. We don't see that in the control conditions. Okay, so what are the physiological implications of this? So we know that the rest is moving from one cell to another through protrusions, also makes protrusions in the organotypic condition. What is the implication? As Kathleen mentioned, Huntington disease, one of the major problems is the striatum degenerates rapidly. Here is a video I wanted to show you. This is a video of MRI images of a patient captured for 4 years, same patient. You can see the striatum degenerates rapidly and also cortex to some extent, but not the cerebellum. The thing is, Huntington disease protein, which is present ubiquitously, why does it affect striatum is not very well understood. So we thought we have been working on this project for many years. What we had a published data showing that the rest converts Huntington into soluble form by adding sumo, and the soluble Huntington become more toxic and causes striatal damage. This was the hypothesis we have been working on. Because rest interacts with Huntington, we wanted to check whether the rest can transport Huntington. That's exactly what we have seen here. So rest, this is a tube, rest-made tube. You see the normal Huntington is not so much present in the tube, but the mutant Huntington, this is the tube, and the mutant Huntington cell, you can see mutant Huntington accumulates in the tube. Here's the video showing that the Huntington can move from cell to cell via this protrusion. So you can see that this is the protrusion, that's where the Huntington being carried to the next cell. Here's another video I want to share with you. You can clearly see the green is rest, red is Huntington. You can clearly see that there's a push and pull. It kind of pushes inside another cell and transfer and delivers Huntington vesicles into the next cell. Here is the quantification showing that there's a dramatic transport of Huntington by rest to neighboring cell. Okay, the question again, the question is, okay, what does it happen in vivo? Can rest transport Huntington in vivo? So that experiment is to address that question, we need to again design new tools. Here, the tool we designed is a dual reporter tool. As all of you know, it's a P2A construct. It makes one RNA, then there will be two proteins in a given cell. So what we did is we took this Huntington, normal Huntington and the mutant Huntington, made them in lentivirus and put them in a wild type mice striatum or as well as rest knockout striatum. This just show the expressions of similar. So we put them in the striatum and then followed them up with time, weeks until a few months. So what happened here is, there is the striatal injected site, Huntington and the reporter, Huntington reporter as well as the EGFP control. They are expressed in similar location in same cell type. And this is the wild type mice. As you can see, we also started seeing the Huntington alone in the other part of the brain. Here it is 500 micron away from the injected site. You start seeing the accumulation of the mutant Huntington. Here is an insert showing there's a beautiful accumulation. This is in the wild type mice. If you do this in the knockout mice, we don't see so much accumulation. There is an accumulation, but it's dramatically diminished in the rest knockout mice. Of course, we do not see wild type Huntington alone being transported from injected site to another, to the other places. There's very little transport compared to mutant huntingtin. Again, the knockout, we don't see much at all. So here is the quantification. You can clearly see that this is the transport of huntingtin from the injected site to 500 micron away, which is dramatically decreased in the knockout, REST knockout. Sorry, this is the injected site. I'm so sorry. This is 500 micron away from the injected site. The injected site, 500 away from the injected site. So we also looked at in the cortex, what happens to the huntingtin in the cortex. And what you can clearly see, here is quantified here, the huntingtin goes to the cortex beautifully. It just moves there, whereas the knockout, the transport is diminished. This emphasizes the point that the REST can mediate the transport of huntingtin or spreading of huntingtin from one place to another. So to summarize my talk, what we think is happening is that in the striatum, there are two kinds of new connections are made by REST. One is a retrograde transport for using this TNT-like protrusion from MSN to the cortex. And there's also collateral connection between MSN to MSN. And one of these structures, REST can be transported itself, as well as transport, in this case, disease-relevant proteins. And I'll show you it can also be involved in transport of receptors and other cargos. So the question is, okay, there are many questions here. How does this tubes are formed, and how they're sustained, and how they are entered into how they deliver the cargo into the acceptor cell. There are many questions we do not know, but briefly, there are initiation step, elongation step, and there is also delivery steps, which we have at least two different mechanisms we think might be happening. Again, there are many open questions here, how this is delivered. So making a tunnel, we all know it's not an easy task. It is a very difficult task. But here, it looks like in our brain, these machineries, they make this constantly, looks like. So maybe there's one making right now in our striatum. We don't know. So here is my team. I wanted to thank the funding support from various organizations. Without them, I wouldn't be able to present this talk. And my research team who have been contributing for all the work that I mentioned. And I thank you for an invitation. Okay.
Thank you, Dr. Subramaniam, excuse me. That was a really nice talk. We do have time for questions if anybody in the audience would like to ask their question live. Please, Oliver.
Yeah, thanks, really fantastic talk. It was just reminding me to the Nature paper from last year it's Ariane Di Polo's lab. She has parasite nanotubes that she nicely shows with in vivo imaging. And I mean, she doesn't show so much so beautifully as you the protein transport, but she definitely has the calcium signaling kind of forming a syncytium of parasites that may constrict the vessels in the neurovascular unit. And it reminds me pretty much to her work, and it looks really beautiful what you have shown. Obviously, you cannot access the striatum in the same way as she can access the retina, so that's a little bit difficult to perform a similar experiment. But I was wondering about your spreading pattern and what else. So you've shown a lot of things in these tubes, and is it sort of, so is the accumulation across the lens, is it, I'm not an expert for Huntington's disease, is this a regular spreading pattern of the protein you're kind of mimicking with those tubes?
Yeah, so that's the point. Like, thank you for this comment. I've seen this Pedicite paper. It's amazing. That paper is awesome. So your question is like, how does it spread pattern? Like, so right now, I think the mechanism we think could be happening. I'm sorry, I went very slightly fast there. So we see that clearly the huntingtin injected in the striatum found in the cortex, and we think that our data supports the idea that it could be happening through protrusions, right? But we don't show that in the, as you mentioned, we didn't access that in the striatum, but we show that in the primary striatal cells, there is a beautiful transport through the protrusion. You can clearly see that. So how does it spread? I think, to me, if you look at simply, it's very, very easy because it just use this pathway and then go into the cortex using this retrograde projection, that there is abundant projection in the projection of cortical neurons in the striatum. It just uses that pathway, existing pathway, but it requires the bridge. That bridge is provided by TNT-like protrusions made by REST. So then it's just kind of easy to move to the other place. If that answers, yeah.
Yeah, sort of. I mean, and then I was wondering, is the same with these nanotubes? I mean, it's kind of it's beautiful when they form, but I mean, those cells, I mean, they're not directly touching with each other. Yeah, I don't know. It's a really great thought. I also would like to think about it a little bit more.
Yeah. Like they do, like if you look at the thing I mentioned, like it is so they do. If you look at this picture, I know I went very fast here. You can see that the tube is touching that. It's delivering and touching. And this is the one puncta which has been delivered already into the neighboring cell. If that understands, it does touch and deliver. But again, I know your point is we do not know the details of how mechanistically it opens up the membrane, or is it a receptor-mediated endocytosis? That's what is happening. We don't know, but you can see that's a beautiful delivery already happened here in this case.
That's fantastic.
So we do have time for maybe one more question.
Andy, there's a question in the chat from Dr. Dietrich.
Oh, thanks. Yeah, sorry, Dalton.
Good seeing you again. Yeah, I do remember that conversation.
Oh, really? Thank you.
Thank you so very much for a really innovative discussion. This is really new to me. So it's a lot of questions based on this new mechanism of communication in neighboring cells. Does neuronal activity affect the transport in any way if you lower or raise neuronal activity, for example?
Yes, this is a great question, Dalton. I think we want to do all those kind of studies. We haven't done those kind of studies because of the. I'm waiting for some funding to kick in. First thing is, and second thing is, those are very important questions. Particularly, for example, REST is induced by thyroid hormone. Thyroid hormone injection increases REST expression in the striatum. So that's a great way to look at it, whether it is an activity-dependent changes. So far, we do not have any experiment done to address those questions. But this is a great question. We don't know yet.
Great. Thank you very much. Good luck with your studies.
Thank you. Appreciate it.
Okay, thanks again, Shri. Okay, our next talk in this session comes from Oliver Bracco, who is an assistant professor in the Department of Biology here at the University of Miami. And the presentation is called VEGF Signaling Causes Stalls in Brain Capillaries and Reduces, sorry, your title's cut off here, and reduces cerebral blood flow in Alzheimer's mice. Thanks, Oliver.
Thank you. All right. All right. Yeah, thank you for the invitation. I'm really happy to be here today and to talk about my work or my past work and a little bit also what we're going to do here. So I just arrived here 8 weeks ago, starting with the 4th semester as a research assistant professor at the Department of Biology. And I'm going to present a just recently accepted story from a student of mine who has been an undergraduate student with me for a couple of years about VEGF signaling and its role in microvascular dysfunction, which is the overall umbrella of my research during the last couple of years, in fact. Oh no. All right. So So as you know, Alzheimer's disease is one of the few major diseases that still has this increase in mortality rate compared to many other major diseases such as HIV, or if you look at stroke, or if you look at certain forms of cancer. And there's currently no cure for Alzheimer's disease, and there hasn't been any medication for the last 15, 20 years, except for this year, where has been one medication, and anti-amyloid antibody has been approved by the FDA, but it's really controversial. But I'm not going to talk about this today. But I would like to say that there's this beautiful study from Ituria Medina, and now it's in Toronto, and where he did look into patients throughout the progression of Alzheimer's disease, starting with some healthy patients and then early mild cognitive decline, late mild cognitive decline, and late onset Alzheimer's disease. Now we just need one graph in this here, which is the vascular contribution, which is the top graph here. So it really emerges early throughout the disease progression and remains important and elevated throughout the disease. And we were particularly interested in one aspect of this, which is the blood flow reductions, which are associated with Alzheimer's disease. And now, here is a study from Haller et al. From 2016, where they used arterial spin labeling MRI to measure tissue perfusion, to measure blood flow changes. You can see here in a normal patient, you can see that the brain is nicely perfused. So red colors are higher perfused and dark colors are less perfused. And you can see that. And if you look at an Alzheimer's patient, you can see there's an overall reduction in blood flow. So there are certain regions which are more impacted than others. And I have a review where I discussed it a little bit about it, but in mice models, we cannot really reflect that. However, overall, we can say that the blood flow is reduced around 10 to 30%, depending on the study and the length of the study, and mouse models also reflects it to one degree. So our question was as simple as, where's this blood flow reduction coming from, and does it impact physiology? And what we typically use is those classic craniotomies on mice. So we remove the skull with a glass window to get optical access to the brain. And then we use multi-photon, so 2 or 3 photon imaging to image the vasculature of the mice. And you can see that here on the right, the typical image, where you have the vascular labeled with a dye. It's called Texas red, so it's labeling the plasma. And here it's a counter stain for methoxyXO4, which also labels protein aggregates. In that case, this amyloid beta. You can see there's not as so much amyloid beta yet accumulated because this has been just a very young mice. Now, to summarize basically all this work in 2 slides, I would like to say that we identified those blocked, obstructed, or we call them stalled capillaries, because it's only transient. And you can see that here. So the diameter of a capillary is smaller than a typical blood cell. So what you can see is a single cell is flowing through the vessel. And here we stain the plasma, but the cells are not labeled. So you can see flow by moving off those black dots along the vessel. Now what we identified, those stalled vessels. So you can see that here on the right, it's a time series, and you see those cells are not moving along, so there's no blood flow occurring at that given time in the cell. So we quantified that for Alzheimer mouse models and did show that this rate is increased about 4 times from a wild type to an Alzheimer mice. Now, to summarize all this work together, because I really would like to present you some new data today, is when we have a stalled capillary, we identified that this is mostly caused by leukocytes, and they adhere or they get stuck in the capillary, and then we don't have blood flow. But when we are interfering with this interaction of leukocyte with the endothelial cell, using an anti-Lysax-G antibody, we could show that this improves blood flow, it improves cognition, and it reduces amyloid beta burden. So when we use the control antibody, that is a non-targeting IgG antibody, we didn't see this phenomenon. Now, of course, our question was, what could be the cause, or what could be the reason of these cells to adhere to the endothelial vessel wall? Now, a student of mine, which has been an undergraduate student with me for 4 years, so he started at the same time I started my postdoc, basically the same day, and with me and working on this project. And with time, he had this idea, maybe those stalled capillaries are places where the blood-brain barrier is leaky. And we know when the blood-brain barrier is leaky, we have toxicity that gets secreted in the vasculature, and we have pro-inflammatory cytokines and other factors that are in the vessel. And maybe this is the reason why neutrophils come in and blocks them. So we have addressed that, and this paper just got accepted 2 weeks ago. So it should be out soon. But I want to talk a little bit about VEGF because it's quite controversial whether it's upregulated or downregulated in Alzheimer's disease. So, however, what we definitely can say is that VEGF can be secreted into the blood from many tissue types and through many routes. It includes the lymphatics, but also the vasculature. And there has been a beautiful study from Craig Brown's lab in Vancouver, where they inhibit the VEGF2 receptor, and that leads to improved clearance of microfilm-induced cortical capillary obstruction. So a similar phenomenon like stalled capillaries, maybe it is even the same phenomenon, but we cannot really tell that yet. However, if you inject VEGF directly into the brain of mice, it improves cognitive function. And our initial idea was maybe we have more VEGF in the brain, maybe also we have more VEGF in the periphery, but the signaling is impaired. And there are some indication in the literature that supports this hypothesis. So I just want to pull that out here, the idea of blood flow reduction caused by VEGF. So you would have cerebral amyloid angiopathy, or you have hypertension, the cardiovascular risk factors contributing to blood flow reduction, which then causes hypoxia in the capillaries, and that leads to increased levels of VEGF, which is modulated by its receptors, and you will get a feedback loop that's regulating. And typically the system would react with more new blood vessels. However, it is known in Alzheimer's disease that you're not really generating new blood vessels, so you have the signaling, but it seems to be impaired. So to jump into the data, so what you can see here is stacks with the 2-photon microscope of the vasculature of wild-type mice, and in Alzheimer mice, we use the APPPS1 model for the study. And in yellow, you can see these are stalled capillaries. So first of all, you see at a given time point, it's not so many, it's 2% of the vessels, but it's transient, and then it's occurring throughout the network. And what you can see here at the bottom is that the stalls are mostly formed by leukocytes, but we also do see all other cell types being involved. We have platelet stalls, we have red blood cell stalls, and we have platelets with red blood cells. Think about a traffic jam, and we have also leukocyte with platelets. However, what was significantly increasing was the number of leukocyte stalls. And we have to keep in mind, although it's only 40 together with leukocytes and red blood cells, 60%, but overall the number of leukocytes may be 1 to 600 compared to red blood cells. So it seems to be that in disease, those leukocyte stalls are really the ones that are increasing. So we designed the experiment having 4 groups, having APP mice for Alzheimer's disease and wild types as controls, either treated with an anti-VEGF-A antibody or with the saline injection. So we injected them every other day, and then we imaged after 1 week and after 2 weeks, and then we compiled the data and looked at histology to identify whether blood-brain barrier leakage is involved in this phenomenon. So first, look in the number of capillary stalls, and what you can see here on the left. So this is a fraction of capillaries that are stalled, and they're increased in APP mice, so in Alzheimer's mice, but with the treatments, they're reduced basically to the level of wild-type mice. And you almost cannot distinguish the two, but we also have to keep in mind that the wild-type stall number is extremely low. It's roughly 0.3%. At a given time when we measure it. Of course, next we ask the question whether this is also impacting capillary blood flow. And for that, we use the 2-photon microscope to image, and with a line scan, we image the individual red blood cells flowing through the vessel, and we calculate the speed, which is the Fourier transformation. And what you can see here is the vessel speed on the y-axis and the groups on the x-axis. And what you can see, you see that there's an overall blood flow reduction in the Alzheimer mice, but the Alzheimer mice that got treated for 1 or for 2 weeks with an anti-VEGF antibody, there's an improvement in blood flow almost to the wild type levels. Now, is it because of constriction? And at least we don't see that at the positions we are measuring. So if we look at the vessel diameter of these capillaries where we measure the blood flow, we don't see a difference in capillary diameter, which is important because there has been recent publications from Andy Shee's lab or from David Edwards' lab, where they're implicating parasite constriction and blood flow reduction. But I think we just have to work out what could be the contribution of each of those phenomena and maybe combine it, which is in theory not difficult, but it's still a lot of data to kind of compile that together. Now, the next question is, what was the mechanism that could be impacted by this anti-VEGF treatment, or at least by the paradigm? So first we measured whether the antibody is really taking out the VEGF-A. So we had brain lysates and we run ELISAs for VEGF-A protein in the Alzheimer's mice. You can see so it's increased overall. And you can also see that there's a reduction in VEGF-A protein levels, in particular compared to the wild type mice. So the antibody is really working. And next we asked which pathway could be involved. So we already have in another project some data on reactive oxygen species contributing to microvascular dysfunction. So we thought maybe we look into eNOS, and eNOS can directly interact with the blood-brain barrier via occludin and via nitric oxide signaling. So we measured the protein level for eNOS, which is the endothelium nitric oxide synthase, so the enzyme that's producing the nitric oxide, and you can see that here. So if we use that with an anti-VEGF treatment, we see a reduction in the protein levels in the Alzheimer's mice, but also in the wild type mice. Now, next we stained for a couple of tight junction protein markers, but I just exemplify that here with the occludin. We were in particular interested in occludin because it's known to be regulated really quick. And we know that stores can be manipulated within minutes, and blood flow can also react to that within 10 minutes or half an hour. Blood flow can change according to the number of capillary stores. So we stain for it. You can see in red, you see GLUT1, which labels just the microvasculature, and occludin is in green. Now, if you look at the wild type mice, you can see it's basically a one-to-one cover between the two. And if you treat them with an anti-VEGF antibody, it remains the same. If you look in Alzheimer's mice, you can see there's a reduced level of occludin because the blood-brain barrier is leaky. But if you use an anti-VEGF antibody, you see a restoration of the signaling. So you really see that occludin is coming back, and with that implicating that the blood-brain barrier is closing again. Now we quantified that for the cortex and also for the hippocampus and could show that this really is increased. The occludin levels are coming back, implicating that the blood-brain barrier is closing again. And now, which I think is a really cool experiment, where I have to admit we spent it almost 2 years, although it doesn't look like it, where we try to map stalled capillaries from in vivo imaging back into the histology. And we tried that with clearing, and we tried this through other ways, but it turned out to be really difficult. But we at least could find some which we could identify some later on again. And you can see one example here. So we took this slice here from this stack, we identified the pattern here in vivo, and then we also found the same pattern again in the histology slice. So overall, we only found 10 of these capillaries that has been stalled. But interestingly, what we then could do, we could measure the occludin level throughout the vessels that has been stalled at that given moment. And what you can see is that vessels that have been stalled have reduced levels of occludin compared to flowing vessels. And this is also true for the remaining stalls, since the antibody doesn't remove all stalls with an anti-VEGF-A treatment. So it's really implicating that the blood-brain barrier is involved in this signaling and in the restoration of the blood flow. Now, as I said, we were really interested in the fast speedness of this experiment. So we designed another experiment where we measure the blood flow in the stalls and the treatment only after 1 hour after treatment. Now, what we did, we have two groups in the longitudinal study. We have the Alzheimer's mice and we have the wild type mice, and we put them on the treatment, and then we image them 1 hour after injection and 7 days after injection. And what you can see here on this picture is that would be the same stack. So at baseline, you can see we have a number of capillaries that are stalled, but if you use an anti-VEGF treatment already after 1 hour, you can see that the stalls are gone. So this is kind of similar what we have seen with this anti-neutrophil treatment. So we knew we are the same pathway, and that was exciting for us. So we also measured and quantified the stall number, and you can see that here. So this is now a longitudinal study, so we could pair the data. And what you can see here is stalls are increased at baseline, but they're reduced in 1 hour and they remain reduced throughout this week, which is similar then to the wild type mice. Now, also here, then we measured the blood flow from the capillaries. It's a little bit more here. It's separated by mice, and it's also separated by individual capillaries that you can see also the real variation in the data because capillary flow is extremely variable. Still in black, we have the average, and you can see there's an improve in blood flow as well, which is roughly 50%, so it's quite significant. And you can see the wild types on the right. Now, same again, if you look at the diameter, there's also a huge variation, but overall, there's no trend for reduced diameters in implicating that maybe on the short scale, constriction is causing the phenotype, and on the longer time scale, maybe it is not as matter anymore, or the other way around, but we did not have any indication for this. So now we looked into the blood-brain barrier itself. And we used the classic assay here, Evans Blue, which I would like to show because it's a little bit more visual. So if you look in an Alzheimer mice, you can see you have some dye that leaks into the tissue because the blood-brain barrier is leaky. It's overall also not as impactful as I thought it would be. But when we treated the mice with an anti-VEGF antibody, you can see you hardly have any leakage left anymore. And so we quantified that. So the experiment has been 1 hour before, and then 1 hour of treatment, and then we sacrifice the animals. So it's this new set of animals. So when we then quantify that in the cortex, you can see there's a reduction of a dye that leaks into the parenchyma, indicating that the blood-brain barrier is less impaired. And if you see it in the hippocampus, we also see a trend, but we didn't see a statistical difference. We also don't have so many animals in this study just because, and we basically only killed them for the Evans Blue assay. So last for this, I really would like to show you some human pictures. So I got human brain tissue from the brain bank, the NIH brain bank from patients and controls with Alzheimer's disease, roughly 75 to 85 years old, which we stained here for lectin. You can see the lectin staining, which labels the vessels. And you can see the occludin staining in red, and you can see a significant amount of the vessels in a healthy controlled patient have still occludin signaling. You can also see that in the overlay. But if you look in an Alzheimer patient, so there's really hardly or really weak signaling only of occludin left. So really implicating that the same mechanisms could be in place. It does not mean it's because of capillary stalling, but at least we could be in the same pathway, and the same impact that we see in mouse could also be true in patients. So we quantified it on the right, but you can see it's not as a dramatic difference as we have seen it in the mice. To summarize that data, and then I'll talk a little bit quickly about what I would like to do here in Miami, is when we have stalled capillaries, mostly caused by leukocytes, but not exclusively. We have this increased levels of VEGF, we have reduced blood flow, and we have the signaling pathway, which is going via eNOS and occludin. At least that is what we have shown. But if you then use an anti-VEGF antibody, so you take away the VEGF from the system, we have improvements in blood flow, we have less microvascular dysfunction, and we have a blood-brain barrier that is closed. So it's really implicating as that this could be a mechanism somehow contributing to the overall vascular inflammation seen in those mice. And interestingly, so just to pitch that a little bit, I would like to say that there has been a beautiful study from Axel Montagne, who is now in Edinburgh, and from the Slockovich lab. They had last year a Nature publication where they were looking into ApoE patients and measured blood-brain barrier permeability, and found out that the dysfunctional blood-brain barrier is predicting cognitive decline. And maybe also this mechanism is contributing to this disease or enhanced disease progression, which we know ApoE4 patients have. So just to put that together is I have spent quite some time, so this is of course a group effort with a lot of people being involved, thinking about what's this microvascular dysfunction or this capillary stalling, where's this coming from? And we know we have Alzheimer's, we have blood flow reduction, so we ask ourselves, is this a chicken and an egg questions which we unfortunately cannot answer, first of all, with the mouse models we are using, but also how the experiments turned out. So I've shown you that blood-brain barrier is involved into this. What I did not show you is that we also have a story ongoing where we look into oxidative stress and reactive oxygen species contributing to this. And I spent it also some time thinking about cardiovascular risk factor that may contribute also to the stalling. Somehow that turned out to be really difficult. It wasn't as easy that if you put a mice on a high-fat diet and you do induce microvascular dysfunction, but it was not so much on the blood flow side. So the blood flow reduction that is associated with obesity seems to be not coming from this obstructed capillaries. The picture remains still complicated and there's still a lot of work to do. So what I would like to do at UM, it's a couple of projects shifting a little bit away from the endothelial cell side and thinking more about the blood cells themselves and the neutrophils. And I'm also would like to look into blood platelets. And so it's really the contribution of neutrophils to dementia. And I really got excited about this project in particular because we know that the neutrophils are originally known for an acute inflammatory response, but it's really showing now that this chronic inflammatory diseases have a strong neutrophil component as well. And all the previous work from us is also hinting towards that, but we really didn't look into those neutrophils. There's some more data, which is really exciting out there, also looking into single cell sequencing of neutrophils in other diseases. And it turns out there are different kinds of neutrophils and some might be more disease associated. So I really would like to also show that in Alzheimer's mice. And then you could think about doing something like adaptive transfer of neutrophils and see whether they induce damage. And I do at least can show some pictures where I have taken out neutrophils from an Alzheimer mice, injected them into a wild type mice. And you can see it here, so it's a little bit weaker. So this is the capillary. So the neutrophils are labeled with a Lysix-G antibody. And what you can see here is over this time series that if you take neutrophils from an Alzheimer mice, put them into a wild type mice, you also see this enhanced level of microvascular dysfunction. So that means definitely the neutrophils are different. And I think it's going to be super exciting to look into this in the future. And now the last thing for the Miami project, I was also thinking there's also exciting data on microvascular dysfunction coming out from other groups, and this is a collaborator from us, which is Evren Erdener, who's now has a lab in Turkey, and David Boas, who did the imaging or is running his imaging lab with OCT imaging, so this optical coherence tomography. So basically, just to make it quick, is like if you see vessels that are moving, you can visualize them, but if vessels are not flowing, they disappear on the picture. It's a little bit like a laser speckle in that sense. And what you beautifully can see after traumatic brain injury is here, so this is pre-injury. If you perform the injury, you see a massive loss of flowing vessels. So they're not degenerating, they're just not flowing at the time because they're stalled by neutrophils, in fact. So if you then follow that over time, you can see the system recovers in roughly a day. For real, it's 2-3 days, and then you're basically back to baseline. And what you also can see, just because it's exciting, is the dynamics of those capillary stalls here. It's a stallogram. So that's an individual stall. You can see some are stalled the whole time. They're imaging, which is 9 minutes. Some are flowing stalled. So you have a lot of dynamics going on, and it's going to be really exciting to see where this ends. To finish this, so first I want to thank, of course, the student who developed the VGF story with Muhammad Ali. He's now at Mount Sinai and continues doing exciting work in patients, and all the other teams that have been involved at Cornell, and now my non-existing lab yet at Miami, but I do have a couple of undergraduate students who are started to learn how to quantify capillary stalls and doing image analysis with Shrindi, Jordan, and Sophia. So really excited to see how that goes and our collaborators and fundings. Yes, thank you.
Thank you, Dr. Bracco. That was really interesting. We do have time for a few questions. Maybe 1 or 2 questions before we move on with the program.
Oliver, this is Dalton Dietrich. Great talk and great to hear your presentation again. Yeah, you're in a really good place. The University of Miami has a very large, as you know, Alzheimer's brain collection. So this chicken and the egg question that you really ultimately have to get to is the vascular perturbation secondary to, neuronal or things of this nature, or primary in some subjects, can be looked at in that. So I wish you well, and looking at, the human tissue along with the transgenic AD mice is a really powerful approach. So really interesting area and exciting area. Thank you.
Thank you.
So there's one. Oh, sorry, go ahead. Yeah.
It's okay. Go ahead. You go ahead, Andy.
Okay, so there's one question that was posted in the chat by James Choi. Thank you, Dr. Bracco, for the interesting data, and welcome to UM. Have you observed whether capillaries with stalled flow are located near plaques or disease-associated microglia? And could you speculate on the major cellular sources of VEGF-A in these tissues?
Yeah, I mean, that's a rough one. So the quick answer is, so we tried to locate, I didn't have this data, so we had the same idea. So the easy one I can answer you, which is no, that I could not localize the position of stalled capillaries to amyloid beta plaques. So we spent a lot of time doing this, but I really couldn't identify it. It looks as seems to be not the case, but the interesting case, it's not random which capillaries are stalled. So if you image them over a long time, it turns out that roughly a third to maybe a half, and maybe if you continue longer, it could be almost all of them. So there are certain, if you want to call them hotspots, that tends to get stalls, and you can a little bit see in this graph as well. So some capillaries are more prone to stalls, and I do have some hypotheses on this, but I don't know. Regarding the microglia, so we looked into microglia, but on an obvious scale, on the macro scale, looking where stalls are. The problem is, as I told you always, is to identify the stalls later on to analyze them post-mortem. So I really cannot say much about the microglia as well. And then it's the same direction. Wait, what was the second part of your question? Next to microglia, and
if you could just speculate on the sources of VEGF-A in these tissues, like where do you think it's coming?
Yeah. So I know that the source of VEGF, whether it's increased or it's decreased, is highly under debate where it's coming from, and I'm not sure I can contribute much to solve that overall problem. Yes, so I definitely think it's coming from the periphery. So I don't have a real proof, but I do see it in the blood, but I also do see it in the brain. So it's a little bit tough to separate that. So my speculation, it's coming from the periphery, but I cannot really prove that as well. And I'm not sure that the APP overexpression mouse model is a good model to do that, unfortunately. So it could come from many cells, could come from the neutrophils as well. It could be many sources. So it's really tough to figure that out. There are some single cell sequencing data on the vasculature itself, and that also turns out to be not really clear where it's coming from. So some of them showing a little bit more on pericytes, and some are also showing it more in endothelial cells. So I'm really not sure where this is really leading us. And it brings me back to my work where I would say, okay, we have the functional impact, whether it's more or it's misregulated, that just needs to be determined. But yeah, I cannot really answer your question here.
That's great. Thank you.
Oliver, great talk. Appreciated that very much. And also all the best for the new exploring in Miami. So my point is like, maybe I missed this idea. So if the stalling is common in AD, is it something to do with, does the patient have a risk for heart attack? Is it anything connected to that?
Yeah, it could well be. So I try to answer this question, which a project which I'm not talking about a little bit, where I look on thrombosis and those capillary stalls. But the quick answer is, so I had the same idea. I put the mice on a high-fat diet and was hoping to see that this really increases the risk. And in mice, it turns out it did increase the risk for microvascular dysfunction and also for changes in blood flow, but it doesn't seem to be as much through this stalled capillaries itself.
Okay. So that means even there is also all the data show that the AD patients have a high risk of heart attack.
Whether this is true?
No. But the AD patients, or the Alzheimer's disease patients, do they have a high propensity to get heart attacks?
No, but I'm not sure actually. But what I do know is it's the other way around. If the patient had a heart attack, it's a higher probability to get Alzheimer's disease. So
okay, that's good.
True.
So
Yeah.
then that means the brain capillary phenomena that you're observing is nothing to do with the peripheral deficit that might happen for some other reason. Okay.
Yeah, and then another ideas which I want to put out, it could be, I think it could have an origin from quite many different sources. So I think all kind of vascular inflammation probably can induce it to a certain degree.
I see. Okay, thank you.
Thank you.
Any other questions for Dr. Bracco? Okay, so if you have anything else, feel free to ask it in the chat of any of the prior symposium speakers. So before we move on to the keynote lecture, which will be the final lecture of this neuroengineering symposium, I'm going to turn it over to my colleague in biomedical engineering, Sarud Rajguru, who will announce the poster awards for the PhD and postdoc poster submissions. Sarud.
All right, thank you, Andy. And again, thank you to all the wonderful speakers today. Really great talk. A lot of new things that we learned. Personally, I learned a lot of new things. So thank you for that. And for the students, yeah, we've received a number of very good presentations. And so the committee of organizers went through all the presentations and decided to award the top 3 prizes. These are cash prizes that you'll be receiving along with certificates from the Institute. And very briefly, I'm not sure that it's probably sharing with you, but let me share it again. There we go. So we have 3 awards. The first one goes to Andrew Cicerillo from Dr. Dumont's lab, who presented a really nice talk earlier on synthetic ligand receptor binding for targeted delivery to the spinal, to the injured spinal cord. So congratulations, Andy. The second place award goes to Kevin Davis from Dr. Prasad's lab, who discussed yesterday on the long-term evaluation of an at-home brain-computer interface in a patient with spinal cord injury. Again, really exciting and interesting data set that I know he's going to continue to expand upon throughout his PhD. So congratulations, Kevin. I know virtually everybody's clapping. I can see Andy clapping, so that's great. And then the third place award goes to Liana Roman from Dr. Manses and Ruggeri's labs, who discussed earlier today, like in the measurements of the mechanical steady state accommodation fluctuations using OCT. So congratulations, Liana, really, really nice presentation as well. Congratulations to all 3 of you. Again, you'll be hearing from Erica and us about the cash prizes that you'll be receiving as a part of that. But also I want to thank all the student speakers who presented, sent abstracts and sent the videos. Typically, we like to have everybody in person presenting their posters so that we can have a lot of great discussions in person. But with the virtual format, at least, like, you know, we're trying to provide a platform for all of us to share our work. So thank you for that. Before we move on to the keynote speaker, I just want to very briefly mention also the new program, a BS-MS program and an MS program that the University of Miami is embarking upon. This will be a program in neural engineering, and the idea is that this will be truly an interdisciplinary program that spreads across all of our different campuses, including College of Engineering, Arts and Sciences, Marine Sciences, and the medical school, and will be involving multiple different academic units in this program. And the idea is really to prepare the next generation of neural engineers who will come in and eventually partake in either graduate studies, go on to industry positions, and lead new efforts as we think about these new technologies, new therapeutics that are coming up in our area. And so as all of you, I know many of us are like, you know, PIs, many of us are students, many of us are trainees in different levels. As you think about it and think about your colleagues and friends, make them aware of it. We'll be sending out more information as the program is established by the end of this fall. We hope to start enrolling students next fall so fall of 2022 in this program. And the degree will essentially be in neural engineering. So this is a very exciting effort that we have, that Dr. Dietrich and all of us have been planning for several years, and eventually it's going to come to fruition. And again, this goes along with other efforts across the United States in some of the leading institutions to sort of synergize these activities together. So very excited about that. Just wanted to turn it over to Dr. Dumont and see if our keynote speaker has joined in yet. I know he is out in California, so it's a slightly different time point, and so we appreciate that. The time difference is a challenge, but I hope he's here and we can.
He's
continue.
here, and we call it sunny California.
Perfect, sunny California. Welcome to sunny Florida.
You got it.
Perfect. Thank you, Dr. Rajguru, and welcome, Dr. Kramer. So it's my pleasure to introduce our keynote speaker today. And we'll be hearing today from Dr. Steve Kramer, who's a professor of neurology at UCLA. He's also the director of research at the California Research Institute, as well as co-PI of the NIH StrokeNet Clinical Trial Network, which is really something that we wanted to emphasize today. He also is the associate editor of Neurorehabilitation and Neurorepair, as well as a section editor for the journal Stroke. Dr. Kramer has spent much of his time in sunny California, where he graduated with highest honors at UC Berkeley. He received his medical degree at USC, and then that was followed by a residency in internal medicine at UCLA, before hopping out to my neck of the woods in Massachusetts and doing a neurology residency, as well as a cerebrovascular disease fellowship at Mass Gen. During his time in Massachusetts, he also earned a master's in clinical investigation from Harvard, something that we'll hear about how much he's put that degree to today. And his research focuses on neural repair after CNS injuries in humans, with emphasis on stroke and recovery of movement, resulting in over 300 manuscripts. A lot of his research has focused on various treatments, with a major emphasis on translating new drugs and devices to reduce disability after stroke, as well as developing biomarkers to improve individualized therapies that meet patient needs. Dr. Kramer has also received the Stroke Rehabilitation Award from the American Heart Association, the Barbara B. Johansson Award in Stroke Recovery from the World Stroke Organization, the Award for Excellence in Post-Acute Stroke Rehabilitation from the American Congress of Rehabilitation Medicine, and he holds the Susan and David Wilstein Endowed Chair in Rehabilitation Medicine. So as you can see, he's the foremost expert in his area. And I'm thrilled that he's going to share some of the exciting work that he's been doing with us today as he discusses brain repair after stroke. So with that, I'd like to turn it over to Dr. Kramer.
Thank you, Courtney. Thank you for the lovely introduction. Let's share a screen here and see if we can make this work. Always a little bit of the unknown. Presenter mode. And let me see now. Normal. Can you see one slide, or do you see all the previous slides as well?
No, we see it in the correct format. Just one.
Fantastic. Slide. Fantastic. All right. Well, thanks everybody. I'll go ahead and get started. I'm going to talk to you today about brain repair therapies after stroke, something I've thought about a lot for the last couple of decades. And we'll have one slide on spinal cord injury, because this is Miami. As Courtney mentioned, along with being at UCLA, I'm the medical director of research at California Rehab Institute. It's a new inpatient rehab facility. It's the largest west of the Mississippi. And just come on out to LA. And if you hit the ocean, you went too far. We're right there in Century City next to UCLA. These are my disclosures. I'll talk to anybody in any hallway about stroke recovery if they can walk fast enough. And this is the way I've broken down the talk today. We're going to talk about three main things. Briefly, I'll review for you a brief summary of the biology of neural repair after stroke. We'll talk about neural repair therapies after stroke, some of the translation issues. We'll talk about some of the issues in clinical trials by way of example and emphasize various principles, such as choosing the target population and pairing a restorative therapy with training. And then key thing is, because injury in the CNS is so different from one person to another. I mean, in the wet lab, often rodents get identical injury one after the other, but in humans, we have to contend with whatever it is that's brought to us, and it could be so different from one person to the next. So we have to figure out which patterns of injury and function and demographics that vary so tremendously across patients with stroke or patients with SCI, so forth. Well, how do you define which characteristics correspond to best responders to a given restorative therapy? And I'll show you some examples from the lab in the context of stroke. And I'll emphasize to you that while I'm a stroke neurologist, and most of the focus here is on stroke, I believe that at some level, an axon is an axon. And if you have injury to a white matter tract or bundle because of whether it's ischemia or hemorrhage or trauma or inflammation, there's probably a good deal of overlap. And so I suspect many of the points here pertain to other forms of CNS acute disease, such as spinal cord injury, traumatic brain injury, MS flares, and so forth. So briefly, let's go ahead and talk about the biology of neurorepair after stroke. Just a couple slides, make sure we're all on the same page. This is my summary of lots of other scientists' work. And the fact is, if you give a rodent or primate an experimental lesion and then check in that brain days or weeks later, you see a whole cascade of restorative events following inflammation, which is the great kickstarter of so many cascades. The growth-associated proteins pouring out, growth factors gushing out at very high levels. It's an excitable brain, and then excess inhibition, angiogenesis, neurons might proliferate, they increase their synapse number, cortical thickness increases. So really going from the gene level to the systems level, histology level, you see a great deal of changes, very stereotyped, characteristic that varies with the lesion. And it's a bilateral event too. In normal brain, there's no such thing as a unilateral brain event unless you have closed genesis. And similarly, in the repair active brain, a unilateral event results in bilateral changes. They may be more prominent ipsilegionally, but they're frequently bilateral. So that's in animals. We also know that while it's not a single biological event, animals have taught us that it's not a single event in time. And so these are data from Claire Holzebach back in the 90s, showing us if you give rodents a stroke, no matter which group they're in, the number of synapses represented by synaptophysin staining increases, peaks at 2 to 4 weeks, and then prunes back. Data from Teresa Jones, 1992, when she was first in Tim Schaller's lab, showing us if you give animals an experimental lesion, that layer 5 pyramidal neurons increase their dendritic branches, peaks around day 18, and prunes back. It's not a static event. Data from Tom Carmichael's lab, my chair, showing that in the 4 weeks following experimental injury to rodents with stroke, some genes turn on right away, some with a delay, some turn on and stay on, some turn on and turn off. And so if you're giving a therapeutic agent, which was a ligand for one of these gene systems, it not only matters what you give, but it matters when you give it, right? So it's a moving 3D target. And in humans, this all adds up to a graph such as the following. This is from Pam Duncan's lab the first time she was at Duke. The x-axis is time after stroke, going out to 6 months. And the y-axis is a measure of motor impairment, named after Axel Fugelmeyer. It's called the Fugelmeyer Motor Scale, where 100 points is normal arm and leg motor status, and 0 points is nada. And you can see that after a stroke, some people have severe motor deficits, some have mild. About 80% have some motor deficits. So it's one of the reasons we study in the lab. It's so common and so impactful on people. And people get better. Everybody gets better after a stroke. Just as everybody gets better after spinal cord injury. It's just usually substantially not enough. But if you look carefully, everybody shows some degree of improvement. And so most of that improvement happens in the first month, perhaps in the first week or two. And then there's usually some change going out to 3 months, especially in the more severe cases. And by 6 months, everyone's at a plateau. In the field, most people think by 11 or 12 weeks, at least 90% of people, 95% of people are at a plateau. And then it's 6, 7, 8 years of survival at a plateau. In fact, and this is outside the aegis of this talk, but by about a year, people start declining functionally. It's a continuous decline as far as people have looked. So it's a plateau and impairment with subsequent functional decline due to a host of factors. some biological, some primarily psychosocial in nature. And here's a slide on spinal cord injury, just a study that caught my eye back in the 90s, just showing how people with spinal cord injury, regardless of whether they're in one treatment group or the other, spinal cord injury sees, I don't remember, 10 or 15% boost in Asia motor scores over the first few months, whether it's in common with stroke, whether it's resolution of edema, or whether cerebral dieschesis is a factor, or whether there is some plasticity that's relevant to motor and perhaps sensory status after spinal cord injury. I'm not prepared to say, but there's definitely a significant spontaneous improvement on average in spinal cord injury, as well as after stroke. So that's just a sort of a summary at the different levels of what happens after focal injury in CNS, in this case due to stroke. So people are working on ways to modify those outcomes so that patients end up with a superior outcome, or at a minimum can get to that same average outcome, but much more quickly, because an accelerated recovery would open other new doors and probably save billions of dollars. There's been a large number of preclinical studies in, like say, a rodent with an experimental stroke, where giving an experimental agent improves long-term behavioral outcome. So these are just some of my favorite examples. This is vagus nerve stimulation, which we'll talk about in a minute. But if you begin stimulating the vagus nerve 7 days after onset of stroke, you see a substantial improvement in the trained task, a supination task in the rodent, compared to no vagus nerve stimulation. What's interesting, not shown in this graph, is if you then switch to a different task, you see generalization, and then follow later, you see sustained gains. So that's pretty exciting. 7-day time window. If you're an acute stroke doc, you're an acute stroke neurologist, one grows weary of running to the ER to meet that 3 or 4.5 hour deadline. So the idea of 7-day time window is most refreshing. Basic fibroblast growth factor. This is work from Seth Finkelstein, my postdoc advisor, showing 24-hour delay after stroke, and you begin intracisternal growth factors. You see a substantial and immediate difference in forelimb placing and hindlimb placing compared to injecting placebo. And Tom Carmichael's shown that a CCR5 inhibitor, Maraviroc, known to much of the world as an HIV drug, an effective one at that, but also inhibiting CCR5 after stroke with up to a 21-day time window, produces substantial and sustained gains in motor status compared to providing the animals with placebo. One of my favorite cells we'll talk about in a minute are bone marrow-derived mesenchymal stromal cells, and substantial data shows that these cells improve multiple outcome measures, even with a 30-day time window after stroke. Of course, there's Dale Corbett's lab, excellent work, showing us how enriched rehabilitation with or without growth factors produces significant gains with a time window that may be no later than 14 days post-stroke. So, I mean, it's a bit of a whirlwind, but just to show you, there are multiple treatment modalities out there with time windows well beyond the injury reperfusion time window measured in hours. So one of the goals that's for the field is how can we take some of these exciting findings in animals and translate that to humans? And that's usually when they call me up. So, Kramer, what do you think? What can we do here? This is sort of my handy dandy rogues gallery list of the categories of therapies being bandied about. There's drugs, biologicals, brain stimulation, lesion bypass like brain-computer interface. Activity-based therapies gets a lot of the spotlight because occupational, physical therapy, speech-language therapy are standard of care, and cognitive therapies. And so I don't have time to talk about all of these or examples in all of these, but I'll touch on a few of them as a means of demonstrating some points and telling you a little bit about what we're up to in the lab. First, to emphasize, and this may be bringing coals to Newcastle for this crowd, but when I speak to neurologists or even to stroke neurologists, they're very caught up in acute stroke therapies. And the idea that there's a repair type therapy as a separate class of therapy isn't always intuitive. So just to illustrate the point, acute therapies aim to promote reperfusion. Stroke is death of cells due to lack of blood flow, which is mainly lack of oxygen, whether it's from an occlusion of an artery or a crack in the artery with an intracerebral hemorrhage or otherwise. Acute therapies try to restore perfusion. And for ischemic, it's a matter of getting rid of a clot. Well, I remind you, a clot is mostly red blood cells, which aren't cells, they have no nucleus, along with platelets. which are not cells, they're just megakaryocyte dandruff, right? With a bunch of fibrin and so forth, and one in 1,000 cells is a white cell got caught up in there. So acute stroke therapies consist of blowing up a acellular jelly ball in order to restore perfusion, either with Drano, such as TPA, or with a stent retriever. That's an acute therapy, and it has to be done within hours, maybe 24 hours in select people. But for the brain, we're dealing with cells and complex circuits. And so the brain is a substantially more complex organ than an acellular jelly ball. And so the fact that the repair therapy field might be lagging a little bit behind the jelly destruction field must be taken in that context. Anyhow, there's a lot of drugs that we could talk about that are in translation that are worthy of consideration. But in order to cover lots of therapies, we're just going to focus on a few of them. In my lab, we're particularly excited about L-DOPA, which I won't talk about further today. The reason is we've come up with a dopamine polygene score that is predictive as to who will respond with better motor learning, better plasticity when given a dopaminergic agent. And I think that's exciting when we can start to personalize restorative therapies based on a polygene genetic score. But let's talk about one of the drug classes that gets a lot of attention in the press these days, and these are the SSRIs. So some of you may recall the FLAME study about 10 years ago. It was an 11-site study in France where they gave 118 people who were within 10 days of stroke and had substantial weakness, either 20 a day of fluoxetine, Prozac, or placebo, once a day for 3 months. These are severe strokes with severe weakness. And the primary outcome measure was this Fugl-Meyer score from 0 to 100. They started with severe deficits around 15. And the gain in the fluoxetine group was 34 points. And the gain in the placebo group was 24 points. And while that's not a cure, a 10-point difference in the Fugl-Meyer scale is something any of us would see across the room. Clearly, limbs are moving better. So this generated a lot of excitement in the field, saying, well, there must be something to this SSRI, whether it's opening critical periods or improving mood and therefore activity or what have you. So some people got really excited about this and designed some other studies, but what they lacked was the following detail. I think FLAME was positive because they picked people who had one thing wrong with them and then followed how that thing changed over time, right? You pick a problem and you follow resolution or not of that problem. And I think that's a good way to design a recovery study. But people got a little bit excited, and what they started designing was, we'll take everybody and follow everything. And I think that's a mishmash, and the heterogeneity will drown out a treatment signal. That is to say, and this is one of the points of the talk, is that the variance between stroke situations vastly outweighs the variance between drug A and placebo, right? So to see an effect, you have to really carefully select the population and match the endpoint to it, because if you just take all comers, you get all kinds of results. So this is one of the studies. It was a well-executed study for what they set to do, and a lot of people worked really hard, but this is what they did. They took 3,000 people who had stroke in the last 2 weeks, and they gave them either fluoxetine 20 a day or placebo. These were mild strokes. Most of them had some motor deficit. So far that sounds all right, a little mild. And they found that fluoxetine had no effect on the primary endpoint. But before we hang fluoxetine out to dry in this context, first let's break it down a little bit. They used as their endpoint the Rankin scale. The modified Rankin scale is the darling of the acute stroke world. It's got 7 levels where 0 is normal and 6 is dead, and 1 to 5 is a little, medium, a lot more problems. All right, so it does not have a lot of granularity. It's a very coarse measurement instrument. And in acute stroke, you can see a difference because when you salvage an entire hemisphere 2 hours into a stroke, you say, well, he was going to go in the very bad area, and now he's in the mild area. Great. You get 10% of people doing that jump with IVTPA, and maybe 30, 40% of people making that jump with reperfusion therapies in carefully selected patients. But to take people who have weakness and stroke and ask to see whether fluoxetine can change the Rankin score, that's a much more difficult thing. You want to have a lot more granularity in your measurement. So they did see that SSRIs reduce depression after stroke. That's a big deal. about 30% of patients with a stroke will experience major depression during the first year after stroke. And as a rule, the literature suggests that clinicians do an incomplete job at recognizing it, an incomplete job of treating it when present. So that's important, but let's go past that to the trial. Thing is, their target population was very broad. They could enroll a patient with any deficit, vision, urinary, weakness, language, memory. I could go on. Anything gets you in the stroke of any severity, from trivial, mild to severe. They could even have a normal CAT scan. So that's a tough way to run a stroke trial because you're just throwing everything to the wind. And so I would assert it's not surprising that we saw these results because I think the name of the game in repair is finding what you're going to repair, measuring the behavioral situation with that injury, and then following that specific behavior and that neural system. But taking anything, everything, anywhere, I think is a problem for the field. Baseline motor deficits were reported as dichotomous, yes or no, which kind of fits with the non-granularity of using that endpoint. And the primary endpoint was measured by having patients mail in a postcard saying, check a box from 0 to 5. They didn't have to check 6 because 6 would be dead, and that's a paradox. So check a box from 0 to 5, how are you doing? And I'm not sure that's the best way to run a stroke recovery trial. Very pragmatic, but we don't do pragmatic trials until we have a positive effect that we want to see if we can make easier to ascertain. And then primary endpoint ranking was not really aligned with any particular entry criteria. And finally, study drug was not paired with behavioral training. And the literature is rife with examples of the following, that neural repair is experience dependent. So this was just a little review we put out earlier this year on principles of neural repair and stroke. And one of them is across different restorative treatment modalities, it's pretty clear that just giving a restorative therapy doesn't give nearly as much bang for the buck as if you give the therapy and pair it with experiential training, right? So the idea is if a kid has a middle ear infection and you give him amoxicillin, he doesn't have to do middle ear exercises. The drug just works. You know, somebody breaks their femur and you put them in a cast, they don't have to sit there and do femur bone exercises. It just repairs. But the brain's not like that. You give a therapy that galvanizes circuits, but you have to have experience to train brain circuits so that they rewire the right way and so forth. So giving an SSRI, but not pairing it with any kind of training probably did not get the maximum effect. So I thought there were a lot of concerns with these very large SSRI trials. I'm hoping that the field learns from these and grows rather than simply labels SSRI as a failed drug and let's move to the next one. That's my hope. Jumping to biological agents, again, we could talk about these for just an hour. Growth factors and monoclonal antibodies, lots of different ways they might promote clinically relevant plasticity after stroke. These are large molecules that don't necessarily get in the brain. People argue about whether some of them get in there when the blood-brain barrier is weakened by injury. There's some evidence that they may not need to get in the brain and that they can exert effects remotely. For example, on the immune system in the abdomen, and resulting in changes in systemic chemokine levels that can modulate neuroplasticity. So that's an exciting area, but it's a long ball because these are hard therapies to try in people. The same may be true of cellular therapies. There are as many cell therapies being looked at to promote stroke recovery as there are cell types in a normal person, it seems sometimes. And everything from induced pluripotent embryonic cells to adult stem cells. Lots of issues about whether they replace brain tissue or nurse injured tissue back to health. One of my favorite cells are adult cells, and I'll tell you a little bit about animal evidence and show you a non-controlled trial we were able to get funded. Was there a question? Did I hear something? Okay, so I've been interested in these cells. I got an R01 to do a trial in 2003, but the FDA said no. And then the FDA said yes in 2007, but NIH said no. So I continue to work towards getting FDA and NIH in the same room so we can do a controlled trial. Once I had trouble getting this funded, I said, well, the pen is mightier than the sword. So let's do a meta-analysis of the preclinical studies on these cells. Mesenchymal stromal cells can be obtained from lots of different body tissues. They're originally obtained from bone marrow. They found that when they chemoablated marrow, for example, in the setting of aggressive breast cancer chemotherapy, that when the marrow reconstituted, there was a particular kind of stromal cell that was just key for successful repopulation of the marrow. And they were able to isolate these cells, they're stromal cells, mesenchymal stromal cells, often from the marrow. And if you take out a little bone marrow aspirate, say 50 or 60 cc's, which may sound ghastly, but clinically it's not much more than a blood draw in the right hands. You take it to the lab, and within a few weeks, you've got enough doses for 5 or 10 people. And if you go huge on it, you can freeze hundreds or thousands of doses from a marrow aspirate if you like cell divisions that way. So we looked in the literature and found 46 preclinical studies of non-modified MSCs. Non-modified meaning they weren't soaked in growth factors, they weren't grown in ischemic conditions, there was no genes knocked in or knocked out, just good old-fashioned MSCs. And what we found is that 44 of the 46 preclinical studies of MSCs for stroke reported that MSCs were significantly better than control. So that's pretty good. I don't think we need to do another 46 studies. I think we're ready to translate them. We have a grant under review now. These results were robust across species, that route, IV or intraarterial, intracerebral. Didn't matter whether they were allogenic or autologous. MSCs lack MHC2, and that's thought to be the reason why they're immunoprivileged. So they don't have to be accompanied by steroids or cyclosporine or any immunosuppressant. You go out to a month, and even the dose didn't matter much. This is one of our graphs from this meta-analysis. This is a forest plot, so-called, because you can see the forest through the trees. And on the right of this ordinate is the effect size favoring MSC, and on the left favoring control. You'll note it's kind of quiet on that. So it doesn't matter whether you give MSCs to rodents or primates with experimental stroke intracisternally, intraarterially, or intravenously, there's a significant effect. It's better when you're invasive, but you don't need to be. And how you want to balance that out is a matter of taste. But across all studies, the effect size was 1.78. And when you adjust for non-published studies and bias, it was still greater than 1. So that's a pretty good effect size for these cells that are pretty well tolerated. This was in 2014. A colleague I collaborate with, Sean Savitz, repeated this effort several years later and found 141 articles with a major benefit for bone marrow-derived MSCs. This is his forest plot, 1.26 between MSCs and controls. There's just a lot of evidence that these things work after stroke. And we're trying to do now is get this into human trials. It's interesting, cardiology and oncology, so many fields have jumped on this, examined it, found it useful or not. But stroke is a little more cautious or slower to the dance. Hopefully we'll get past that. So while trying to get a properly controlled blinded trial underway, a company in San Diego approached me. They had a GMP facility that makes very clean MSCs, and we got FDA approval to do a non-controlled trial. They did not want a control group. They wanted to give their cells to 36 patients with severe impairment and see how safe they were. So this was published in Stroke a couple of years ago. It was safety and preliminary efficacy of IV allogeneic MSCs in patients with chronic stroke characterized by severe impairment. NIH stroke scale score had to be greater than 6, which is a lot of impairment for chronic stroke. We gave them 1.5 million cells per kilogram body weight, which is sort of a low medium dose for the field. And we found it to be safe. And over 12 months, we saw significant gains in every measure that we looked at. NIH stroke scale, Barthel index of function, mini-mental status, and geriatric depression. I want to remind you that these patients were average of 3 years post-stroke. At 3 years, there is a net decline in function year after year. So these patients were getting better when most patients should be declining. Now, you tell somebody with impairment, I'm going to give you some stem cells. Let's see if you get better. There could very well be a large placebo effect. But just realize that the context normally would be one in the opposite direction. Exciting, when you look at the Barthel score of 95 to 100, which is just described as an excellent functional outcome, it went from 11% at baseline to 35% at a year. So I think there's a signal there. And a study like this needs to be repeated with a proper control group. There aren't many animal studies for chronic stroke, and how many months post-stroke in a rodent corresponds to 3 years post-stroke in a human. I know these are complex issues. So this 3-year human study wasn't built on a great deal of literature in animals late after stroke. It was built on the literature giving MSCs within a month of stroke in rodents. So this line of research might benefit from some specific preclinical studies bolstering the benefit or characterizing the effects of late benefit in humans with stroke. But this is a good start. It's something that underscores the value of continuing with these cells. And when we talk about brain stimulation, I mean, there's many ways to stimulate the brain, from stimulating the CNS to stimulating the peripheral nervous system. You know, the whole thing runs on 1/8 of a volt. But there's a lot of range to play with in order to change neural function. TMS, transcranial DC stimulation, get a lot of airplay, and they're showing some effects. It's not quite clear yet what the benefit is of those interventions and which stimulation paradigm to use, unilateral, bilateral, high frequency, low frequency. It's not clear which population is most likely to benefit. It probably varies across stimulation modalities. It's also not clear what kind of behavioral training should accompany brain stimulation. It's a young field and there's some exciting leads, but nothing that's cinched it yet. Gottfried Schlaug at UMass and Wayne Feng at Duke have a large trial going on in StrokeNet called Transport-2, where they're looking at a bilateral TDCS approach paired with intensive constraint-induced motor therapy for patients with subacute stroke and arm motor deficits. Anyhow, we could talk about brain stimulation for an hour, but I thought we'd talk about one recent result from a team that I was part of that's intriguing, and that's vagus nerve stimulation. Remember, vagus comes from I think it's the Latin root for wandering, same as vagabond, right? So vagus nerve covers all the thorax, all of the abdomen, and most of the pelvis. And so over millions of years, the brain has learned to take vagus nerve afferents, parasympathetic afferents, to heart. That's weird, brain to heart. The brain listens carefully to vagus nerve afferents and has rapid plasticity based on vagus nerve incoming data because parasympathetic data is life or death, right? So Michael Kilgard, who had worked with Mike Merznick, set out at UT Dallas to look at how well vagus nerve stimulation might relate to plasticity. And his first study was in the auditory system, and that got a lot of good press. And then he focused on pairing vagus nerve stimulation with movement to look at motor system plasticity, and then with motor system plasticity after stroke. So I started working with this team in 2011 when they were first planning their motor system studies. And we did a series of studies, first three studies looking at vagus nerve stimulation after stroke in animals, and they all looked pretty good. And this led to two early phase human trials looking at the safety and feasibility and preliminary efficacy of pairing vagus nerve stimulation with motor training in patients with chronic stroke. And so what happens is the patient has a vagus nerve stimulator implanted in the neck here. It's a pretty simple procedure if you're an extremely skilled surgeon. And then they tunnel a little receiver under the chest like a pacemaker. And when the patient goes to make a movement with the arm, the therapist pushes a button that wirelessly sends a signal to the vagus nerve. And this improves arm function compared to no vagus nerve stimulation and the same intensive motor training. And so that led to this study that was published in Lancet earlier this year, which was meant to be a pivotal trial. It's double-blind, sham-controlled, randomized trial. Now, 19 sites over a couple of years to find 108 patients. So it's a carefully selected population. We saw a signal, as you'll see, but how much does it generalize? One can ask. Patients with chronic stroke and on a weak arm. This is a good control. In surgical trials, one has to ask, how aggressively does one go for the control arm? And a mild version is to not do surgery or to just cut the skin. In this group, everybody got a VNS implanted, a vagus nerve stimulator implanted. This is the same surgery, the same device we use for treatment refractory epilepsy or depression, also approved for OCD. So in this case, we put a VNS in everybody and turned it on in half the patients with rehab versus surgically implanted, but never turned on plus rehab. So that's a very aggressive control. And that's good. It gives you confidence that it was the stimulation itself and not the expectation bias that patients were getting a surgery. Oh my God, I'm going to get better kind of thing, which is real, of course. And so they got 6 weeks of intensive daily therapist supervised therapy with VNS turned on or not, followed by another 6 weeks or so of in-home self-administered therapy. About 400 simulations with each treatment session. And for the shock jockeys, it was 0.8 milliamps at 30 hertz. The main result was this: the Fugl-Meyer, this is the arm Fugl-Meyer, 66-point scale. It went up 5 points in the VNS group and 2.5 points in the non-VNS group. So on the one hand, p-value is significant. People who got vagus nerve stimulation had greater motor gains than people who didn't. True. So the study met its primary endpoint. But is a 5-point Fugl-Meyer gain out of 66 clinically important? It's right at the edge of what the literature says is the minimal clinically important difference. But even then, the Fugl-Meyer scale is neither linear nor continuous. So it's hard to know what that means, MCID in this context. But so what I say is there's a modest gain. Moreover, the control group that got rehab therapy with no vagus nerve stimulation went up 2.5 points. So now we're talking about a 2.5 point difference between active and inactive VNS. And that's really getting a modest or even mild, the difference. So if you had a patient who was just starting stroke recovery, you might say that's an aggressive surgery for a low bang for the buck. If you're somebody who's 3 years out and wants to try everything, you might say, I'll take that chance, right? Another thing to realize is that if you followed them from 6 weeks to 12 weeks, the difference spread a little bit. Okay, I'm getting a little more confidence. If you look at a responder analysis where we took a cutoff for interesting amount of Fugl-Meyer point gain, I think it was 6 points, that you saw that twice as often in the VNS group as in the placebo group. So that suggests that there's some people who respond really well, and there's a lot more of them in the active group than placebo, right? So it may come down to finding out who are the responders. And when we looked at a functional secondary outcome, the Wolf Motor Function Test of arm function, the responder analysis showed 2.5-fold more people in the VNS group. So I see this and I say, there's a signal here to neural stimulation. There's a signal to VNS that there's something going on that's better to have it than not. And if we can find the right group, maybe we can spread this responder analysis to everybody and have the difference be larger in a larger population. And a nice touch for the study, 18% of the people getting VNS thought they were getting VNS. 18% of the control group thought they were getting VNS. So it was well cloaked. There was no expectancy bias, right? Lots more we could say about brain stimulation, but we'll jump on here. Then there's lesion bypass. I won't talk about this, but what an exciting thing it would be to peer into the future 100 years from now and see how machines are interfacing with the human brain if we're still around. But right now, when we talk about brain-computer interface, we usually mean one of two things: either a fairly aggressive surgery where there's implanted hardware that's indwelling, sometimes for a year or two, but it seems a little aggressive to the common person to have a bolt sticking out of the head at all times. Or we have non-invasive. We do some of this in the lab where we have, for example, an EEG that detects certain large-scale clunky brain events and then uses that to control an end effector, right? So the idea is that you record signals from intact brain areas that have been de-efferented and can no longer control muscles or other body parts. And you record signals from intact cortex, for example, and use that to drive an exoskeleton or, in our case, a functional electrical stimulator. So this is a promising area, still maybe at the early stages for large-scale clinical application. Then we'll talk about activity-based therapies for a few minutes. As I say, they're part of standard of care. A constraint-induced therapy, the EXCITE trial published in JAMA in 2006, is one of the few positive phase 3 trials in stroke recovery. 2 weeks of intensive boot camp, 6 hours a day, for people with arm weakness after stroke, produces gains that are much greater than those in the control group, and they are sustained at 2 years. So that's exciting. A lot of robot therapies are just there to guide repetitive therapy, right? Guide repetitive movements. And then there's telerehabilitation. So telerehabilitation is a form of repetitive activity where it can be done in lots of different settings. And I'll show you some of our data, why I'm kind of pretty gaga about this. First, it's important to emphasize that activity-based therapies have value in 2 ways, building on the slide of the principles of neural repair that I mentioned before. The first is that activity-based therapies can improve outcomes. And we're going to look at that in a minute. Second is that they're sort of an adjuvant, if you will, to many of the other types of restorative therapy, experience-dependent plasticity. So if you give somebody a drug to improve aphasia or arm weakness, you probably want to pair it with language therapy or occupational physical therapy, respectively. Right? So they do good things on their own, and they're a key adjuvant to many of the other therapies you see on the screen right now. So why do we talk about telerehabilitation? So we had done some robotic studies in the lab when I landed at Irvine, where I was at for many years before UCLA, I was fortunate enough to collaborate with David Renkensmeyer, which is one of the world's top robotics engineers and an exciting guy to talk to, really neat guy. And so some of his students collaborated in my lab, and we were building robots for arm movement. And in the process, we wrote a lot of games because we gamify it to keep it interesting. And we were having people do 2,000 repetitions of hand movement a day. That's a lot. And we showed some gains, and we were able to characterize predictors of treatment gains, but patients had to come to us every time. And that's not always practical. So we thought, how can we get these games and high levels of repetitive movement into the home? And so let's try telerehab. We were doing this well before COVID, and then it became topical. The reason we're thinking about this is you got to get a high dose of practice in to change the brain, right? Animal studies suggest you get favorable motor system plasticity with 600 to 700 repetitions per day. And humans, we know that higher doses of rehab therapy can improve outcomes. A meta-analysis by Keith Lowe's et al. The x-axis is number of hours of active therapy provided by a clinical trial. Each dot is clinical trial. And the y-axis is the effect size, HSG. And you see that the higher the dose of rehab therapy you give humans after stroke, the higher the benefit. It's a pretty good fit on average, a couple outliers. So higher doses are better. Why? Because you get more of this. This is one of our robot studies. And we did a functional MRI during paretic hand squeezing. Before therapy, you see a little bit of paretic stroke affected side, primary sensory motor cortex activation during hand squeezing relative to rest. And then after a few weeks of intensive daily therapy, a lot more activity during the same movement. And so this is the kind of thing that we're trying to promote. But people don't get a lot of therapy. They don't get intensive therapy. They don't get 800 a day. They don't get thousands of repetitions a day. Because it's expensive, nobody wants to pay for it. It's hard to access. It may be that somebody lives 30 miles from the nearest physical therapist, or it may be like in Los Angeles, where there's a good therapist 2 miles away, but you don't have an hour to get there. People are not compliant with assignments. So even if they have access to everything, people don't listen to their clinicians. They take their drugs with about 50, 60% compliance in the US, and same with a physical therapy with or without a stroke. And then even in optimal conditions where people can access it and are compliant, the dose of activity-based therapy provided in stroke rehabilitation is really modest. Catherine Lang had a landmark study in 2009 where they actually sat and counted what the therapists were doing, and patients were getting 32 repetitions per session. I'm at 4, I'm at 5, and multiple studies have confirmed this. And the reason is that during the 45 to 60 minutes allotted for OT or PT, therapists have to examine the patient, review the chart, talk to the patient. There's a lot of psychology in good OT PT is what I'm seeing. And so they have to walk them through it. But if you want to change the brain, you have to practice a lot of movements. So for example, I play French horn in the LA Doctor Symphony Orchestra. Come on by sometime, we'll do a show for you. And I assume you don't play. And if I gave you a French horn and said, play a nice clean C major scale, and you practiced it 32 times, you'd probably sound pretty stinky. And if you practice 32 times a day for a few months, you probably sound pretty stinky. But if you did 2,000 scales a day, it's not hard to see why lots of practice can make for better motor system plasticity and motor function. And it's not just the quantity of rehab that sometimes can be wanting. The quality of rehab is also important because it's well known through the literature, stroke and non-stroke, that you get greater brain plasticity when a practice task is challenging, interesting, motivating, relevant, right? So how do we get better quantity and quality of motor therapy after stroke? And our answer was telerehabilitation, which has been defined as delivery of rehab services via information and communication technologies. Importantly, and I'm learning this more and more working my lab now housed in an inpatient rehab facility, that rehab medicine is much more than just activity-based therapy. Maybe obvious to some, but includes assessment, monitoring, prevention, supervision, education. And so that's the kind of system we set out to build for telerehab. This is one of our more recent systems. We deliver to the patient's house a table and a chair and a computer that's connected to the internet. And the computer has attached to it all kinds of little doodahs that capture various aspects of upper extremity movement, proximal, distal. For this is supination, pronation. You can hold PlayStation Eye and it gets you off the table, and that's very proximal movement. We have pinching and tapping and a plastic pistol because people like shooting guns. They like shooting ducks a lot. People really like shooting ducks. So, all right, we'll let them do it. And then there's a video camera because socialization is just critical. This may be one of the more useful slides to share with you today. We consider these to be maybe our 5 most important core principles as to why our telerehab isn't working so well. First off, it has to be easy to access and easy to use. There's no off-on switch. There's no hitting any key. In fact, we take the keyboard with us. It has to be in the house, and you just hit a button to begin, and it has to be that simple, right? We promote high-intensity movement practice, over 1,000 repetitions per day rather than 32. And it has to engage numerous brain circuits. So our games are not just simple games, but we use visual motor tracking to bring in intraparietal sulcus, premotor, primary motor circuits. We use read the rules, follow the rules, and then decide which movement to bring in prefrontal, dorsal premotor, motor activities. We use sensory motor processing to bring in, postcentral gyrus activity, and so on and so forth. Lots of different ways to have M1 fire, and we try to attach to many of them. And then gamification, because games are fun and exercise is not so fun to everybody. And then maybe the most important thing is that the occupational and physical therapists who administer our telerehab, socialization and supervision. So a licensed therapist supervises every other day, but during doing so, there's socialization, which is just pivotal. So here's Lucy having a conversation with and watching this patient play whack-a-mole, which is fun, by the way. And socialization is just part of the secret sauce there. It just gets people doing it and enjoying it. This is what they see. They start, it says, press the big green button to start. We have verbal and nonverbal instructions at all times for the right and left hemisphere intact, right? And this is a movie of a hand going up and down, so I kind of get it. And there's a big green button, boom, and they're ready to go. And then the first task comes up, whatever the therapist assigned, and they finish that, it comes back and the box is checked, hit the green button to start the next one. In this case, Flappy Bird, right? These are some of the main ones we have. We have driving games. Everyone likes to drive. As I mentioned, people really like shooting ducks, so much so we have a second duck shooting game. Space Invaders, which tells you when I came of age in the video era. Everyone likes playing music. Gambling games are the most popular. We have slot machines. So the reels are spinning and you have to hit a button to make the timing of the reel line up. So you have the same thing, right? So you're looking at timing. It's a very cerebellar, corticocerebellar circuit. We also have video poker, which is just 1 hertz movements, and blackjack, solitaire. We have exercises now in video format, over 100 arm exercises. We do lots of assessments, Likert scales, and we educate people with stroke jeopardy. People like that. But the one people like the most, we did a qualitative study, is the video conferences with their therapist. That's the part that they really like. We did some pilot studies that were favorable, which I don't have time to talk about today. But then we were funded in StrokeNet to do an 11-site study. So StrokeNet, as you may or may not know, is the NINDS Clinical Trials Network for stroke. It's been going on about 7 or 8 years. And Walter Karashets, who was my residency director, is now head of NINDS, and he appreciates that repair is important. So there's the acute stroke pillar, there's the preventative pillar, and now there's also a recovery repair pillar, and that's the one that I head there. On a side note, just advertising, if you have an idea for something that looks good in animals and for which there's at least some positive pilot data in humans, and you're ready for a multi-site study, send me an email. I'm the only Steve Kramer at UCLA, and we can talk about getting whether your idea is appropriate for a large-scale study, and if so, moving it forward. Anyhow, this was the first StrokeNet recovery trial, and we took the thing we had done in a single-site study and said, let's do it at 11 US sites. It was 124 patients, 1 to 9 months after stroke, and they had arm weakness. They got 6 sessions a week for 6 weeks, 36 sessions, each 70 minutes, either in the clinic, where Monday, Wednesday, Friday, they had to drive to a clinic, and Tuesday, Thursday, Saturday, they used a printout and did their exercises at home, or everything done in the home, where Monday, Wednesday, Friday, they did telerehab with a therapist supervising, and Tuesday, Thursday, Saturday, they just did the telerehab on their own, in the home with no therapist supervision. And it was a non-inferiority design. So we hypothesized that 6 weeks of intensive daily arm motor therapy provided in traditional in-clinic format would produce substantial arm motor gains, and that dose-matched, intensity-matched therapy using telehealth methods would produce identical gains, that is, gains that were not inferior. And that's what we found. Both groups went up by about 8 Fugl-Meyer points, which is pretty good. And when you adjust for pre-stated covariates, the difference between groups was 0.06 points in favor of telerehab. That's a trivial difference. And the 95% confidence interval was inside the non-inferiority margin. And so we say that telerehab was not inferior to in-clinic therapy. Presence of aphasia had no difference on treatment gains, which is nice. And we were doing over 1,000 arm repetitions per day, which is much higher than 32. So our conclusions from the study was that home-based telerehab after stroke is clinically and statistically significant for improving arm function, and the efficacy was comparable to in-clinic therapy. So that's sort of a whirlwind review of some of the neural pair therapies being translated, maybe some of the key principles therein. In the last few minutes, let's just talk about some of the work with biomarkers. We'll focus on work from my lab, but there's a lot of work going on here. These therapies don't work the same in everybody, and the efficacy signal is diluted if you give a treatment to somebody who is unlikely to respond to it. So one of the challenges is, and no different from many other, no different from pulmonary medicine, no different from oncology, how do we find the people who are most appropriate for a given therapy and connect the right people with the right therapy? Personalized medicine is the word of the day in neural repair therapies. So we're looking for biomarkers that are validated, that stratify people, that break them into different subgroups. And what we found in our lab repeatedly is that measures of neural injury and neural function are often key and additive. Neural injury makes sense. Big injury, hard to improve. Big injury to the motor system, hard to improve the motor system, right? But neural function is something that is not well entrenched in clinical neuroscience. And the idea being not to measure the behavioral phenotype at the bedside, but to measure the activity, the functional activity of brain circuits as a determinant of their capacity to undergo plasticity. We have a lot of ways we can measure neural injury or brain structure clinically. I can order a CT, an MR, et cetera, but we don't measure neural function often. Again, apart from its surrogate, the behavioral phenotype, the downstream expression of neural function. You can do a PET FDG scan, to which I respond, go ahead, you can do it. We do evoke potentials during an MS workup, but we don't do a lot. There's functional MRI and connectivity emerging in the research literature, but these have not really made strong ingress into mainstream clinical practice. So kind of a research maybe leading the way a little bit. So I've been talking about stratifying for repair for a while. If you do so, it just cleans up your sample and you get higher power, lower cost, shorter study duration, et cetera. Of course, I'm not the first to think of this. One of my heroes, Axel Fugl-Meyer, the great Swedish physiatrist, 1982. It suggested that in patients found suited for admittance to rehab wards, visualizing the brain damage may be a useful adjunct. So I just love this quote. And we still don't do that. If somebody has an acute stroke and they're admitted to the hospital and they say, should we send them to the inpatient rehab facility or not? It's all based on wallet biopsy, chart review, and bedside exam. There's nothing about the CT or MR yet, and nothing about the measure of neural function yet that influences the decision, the triage decision, is whether they go to a nursing home or inpatient rehab. But I do think that will change. It might take a few decades, but I think we'll figure out what the answers are and they'll be put into clinical practice. So I'll illustrate some of these points using data from a trial we published a few years ago, where neural function and neural injury were both found to be important predictors of treatment effect, this in late subacute stroke. Let's talk about injury first. So here's two patients in a robotic study. They're both a few months post-stroke. This guy has a big stroke. This guy has a little stroke. I put it at red arrow, shows you where the stroke is. Can't miss it. They have similar substantial motor deficits. Arm Fugl-Meyer score 20 out of 66, 21 out of 66. So same deficits, different size stroke. Same impairment, they get the same therapy. Who's going to do better? Well, obviously the guy with the little stroke, 6-point gain. Big stroke, 1-point gain. So all you have to do is measure the infarct volume and you know who to put in your trials, who to send to rehab, right? Well, big stroke, little stroke, same baseline impairment. Who's going to do better, right? Same impairment, same therapy. Well, in this case, the big stroke had a 10-point gain with identical robot therapy. This guy had a 2.5-point gain. Why? Probably because the large stroke spared the corticospinal tract, which you can see coursing through the centrum summa valley through the internal capsule here. Whereas this tiny little stroke nailed the corticospinal tract. and that's a big deal. So that's what lacuna strokes like to do. So it's not just a matter of measuring the injury, location, location, location. It matters where the injury is for answering questions like who's going to respond. So we can use diffusion tensor imaging tractography to outline normal white matter tracts and superimpose different infarcts on them. And from that, determine the percent corticospinal tract injury. The higher percent corticospinal tract injury, the less I can make people better with an intensive arm motor rehab program. And percent corticospinal tract injury is a better predictor of treatment effect than total infarct volume, baseline deficits, age, time post-stroke. But we're still only explaining a third of the variance. So we look for other things. And so we look at neural function. And we find that different measures of motor system connectivity here between ipsilesional and contralesional motor cortices, this is also predictive of capacity to undergo improvement. The model being that a healthy level of connectivity between the two hemispheres is associated with a higher capacity to improve with therapy. And in the same study, greater CST injury is bad, less M1-M1 connectivity is bad. These two measures were independent and additive in a multi-variable linear regression model for predicting treatment response. So we're up to about 50% explaining the variance with these two measures. Of course, measuring brain function is tough with MRI. It's not mainstream. And you have to take somebody to the basement, put them in a soup can, tell them to hold still. 10% of people would rather die than go in an MRI machine, really. And so maybe we can do this at the bedside. So I don't have time to get too much into the EEG data, but we have a 256 lead saline lead system we got from EGI in Oregon. I think they're owned by Philips now. And the impedances are a little higher than gel, but they're still plenty fine to ask questions of our signals. It goes pretty quickly. Our prior model was using resting EEG. Now we're doing event related. But in one of our earlier telerehab studies, in EEG at rest, prior to therapy, prior to patients even seeing the device they were going to use, we just listened to the brain at rest in chronic and subacute stroke. We asked who has connectivity, coherence with ipsilesional primary motor cortex, that's C3 and the surrounding leads, in the high beta 20 to 30 hertz range, which is sort of the language of much of the motor system. We found that the more that stroke side motor cortex was coherent at 20, 30 hertz with contralesional motor cortex, with ipsilesional prefrontal dorsal premotor, with some superior parietal stuff, the more there's connectivity there at baseline, the more people are going to improve with a subsequent 4 weeks intense telerehab program. And here we're able to explain 61% of the variance. I don't have time to get into genetics, but we've been looking at polymorphisms and polygene scores and whether they also have a seat at the table for identifying treatment responders. It's complicated. There's a lot of genes out there. We just finished a STRONG study. We followed 600 patients from acute stroke to 1 year into detailed behavioral phenotyping. It's not a large sample for GWAS, but we are going to look at a number of candidate polymorphisms to see if genetic variation explains differences in outcome in terms of functional outcome, motor outcome, stress outcome, and a number of related factors. All right, so that was sort of a whirlwind tour. We talked about the biology of neural repair. We looked at some of the things that work in animals and how translation is going in humans across a number of different treatment modalities. And we considered a couple of principles, such as importance of selecting a target population, pairing with training. And we looked at different biomarkers that might help us identify the target population, such as measures of neural injury and neural function. And with that, I thank you for your attention today.
That was a really great talk, and I appreciate that you covered so many different areas, so thank you for that. I want to open it up to questions from the audience. You can either unmute yourselves and pose your question verbally, or you can type it into the text or the chat box, and I can read it for you. The choice is yours. Dalton, I think you need to unmute.
I am sorry. Stephen, thank you very, very much. This is Dalton Dietrich for contributing to our neuroengineering symposium. We actually had a very nice talk yesterday morning on vagal nerve stimulation. And so we're excited about that particular approach as you are. I was wondering in your MSc chronic stroke study, what do you think the mechanism is in terms of improving function? And would you couple a chronic stroke study with a cell therapy with rehabilitation?
Yeah, it's a good question because, as emphasized, it wasn't really built on preclinical data in the chronic phase. So one might assume that some of the acute or subacute rodent work pertains. By the time you're in the chronic phase, my guess is it's more of an immunomodulatory therapy than anything. And that may be the effect in acute stroke too. It's getting a little bit out of my expertise. Anecdotally, one of the patients who did pretty well, he had a clear-cut jump in function. He was on Enbrel, one of the drugs for rheumatoid arthritis. And he skipped it for 6 months because he didn't need it after getting the MSCs. And then he needed it again. And I can't say that was correlated, it's anecdotal, but I suspect that these cells go in, very few of them get to the brain. A lot of them get caught up in the spleen. I suspect it's a systemic immunomodulation, which could have downstream plasticity effects to my knowledge, but it's probably a systemically mediated effect. It would be fun to get in there and check blood levels of some of the chemokines and see which of them correspond. This company had a very targeted goal. So those are the data.
Yeah, one of our interests in the lab is looking at the effects of stroke on other organ systems. And it's really interesting how focal stroke can actually, and focal trauma or spinal cord injury can actually affect different organ systems, and including the spleen. So you could actually visualize that the stroke is affecting the phenotype expression of various inflammatory markers that are floating around the whole body and could be affecting the ability of the brain to respond to some type of reparative strategy. So really interesting topic, and thank you again for participating. Great talk.
Yeah, thanks. Yeah, it's fascinating. Gearing up for an MSc grant, we actually looked at the spleen in 100 patients after stroke and found that no MSCs, splenic volume contracts about 20-30% by day 3 and then expands back out. Sean Savage showed it with ultrasound, and we showed it with CT. I bet that's part of the target. This is Patty Hearn's work, to my knowledge. And it may be the whole effect of MSCs, although there's probably something in the brain directly to the edge of the infarct, if I can read the literature.
I was also interested in the MSC work in particular because there was a recent clinical trial for acute respiratory distress syndrome associated with COVID here at the University of Miami, specifically, I believe it was run by the Diabetes Research Institute, and they were actually using umbilical cord MSCs. So to your knowledge, is there any distinction in stroke between the various sources of MSCs? I know your work was focusing more on bone marrow derived MSCs.
I don't think I have the expertise to answer that. To my superficial knowledge, whether you get the cells from umbilical cells or marrow or dental pulp or adipose tissue, they all seem to work. But whether there are more refined comparisons, I'm not able to answer. But they all seem to do pretty well.
Yeah, thank you. Are there other questions from the audience?
I was going to ask, and then your last slide basically talked about it, genetic predisposition to stroke, and therefore just followed up genetic factors that could be involved in reparative strategies. Ralph Sacco is our chair in the Department of Neurology, and he's been looking for many years at predisposition of stroke and genetic factors with different populations. But now it looks like more and more interest is getting into how genetic factors actually interplay in terms of ability to recover from stroke or any other types of brain injury. And you touched upon that. But I was really interested in these very large stroke versus small stroke and how you get this, just depending on what the anatomical area is that's affected. And I guess that's where functional MRI and things like that is really helping us in terms of patient heterogeneity questions.
Yeah, but it's got to be, it just makes sense that the core mechanisms of recovery from stroke would differ between a small white matter stroke versus a stroke that destroys a lot of cortex. A stroke that affects corpus callosum versus a stroke that spares it. A stroke in a 20-year-old with a robust immune system versus a stroke in an 80-year-old where everything is atrophic and fibrosed. And I mean, it's complicated. One hopes that there's enough in common that there's a treatment that can help some subpopulation of them. You know, my efforts to date focused on polymorphisms, you know, the ApoE4 polymorphism and a common BDNF polymorphism. We found some differences in healthy subjects and some preliminary results in stroke, but it hasn't really panned out yet. Some excitement, some disappointment. But now we're looking at we have like 30 main hypotheses in this strong study that I mentioned. And as I say, I'm particularly interested in the dopamine system, which probably is in common to a lot of these strokes, as well as polymorphisms in some of the common growth factors. That's just based on some animal literature. But yeah, if there was one mechanism for stroke recovery, you could say, well, let's look at genetic variation in it. But there's so many players, and those players differ according to key variants in stroke, including injury size and topology, but age and other stuff too. So it's a big problem, but you know, we're chipping away at it, one hypothesis at a time.
Steve, I would have a question. So I have two questions for your tele-rehab, which I found really exciting. But you found in the end, which I think is also interesting, that there wasn't a difference in the quality of the training between the two, but I wasn't sure, is the repetition really higher if they do it at home? So, you know, you said the number of repetition versus the training, the ones that who came in to the physiotherapist to do the training. Right. So because if it would have been different, my interest would have been, it didn't make then a difference, just the number of repetitions, or maybe the social aspects, the interaction with the psychotherapist, with the physiotherapist made the difference to be
right.
equal in the end.
It's a good question, and I don't have the data to answer it. So in the people who got tele-rehab, we took a convenience sample and counted the repetitions every day, which is racking labor, and we averaged at 1,031. For the people in clinic, we didn't count. It was actually sort of a last minute kind of thought. So we actually didn't build that in prospectively. If I had to guess, I would say in clinic, they were getting less. We did structure it. So in clinic, they also had to have 1 to 3 minute little exercises and 70 minutes of it. So we really tried to match the dose and the intensity. So I suspect it wasn't too far off, but I don't have the data to back that up. You know, in clinic, just to...
Yeah. Can I also speculate? I guess you don't have the data. Obviously, you probably don't have the data, but do you have a comment on post-stroke dementia and motor rehabilitation, which I was also once interested in, whether you recover from your motor dysfunction to a certain degree, but then you still have this increased risk for dementia later in life? Do you maybe have some comments on that?
Right. Now, this is me speaking not as a researcher aware of the literature, but as a clinical stroke neurologist. We know that a regular exercise program, including an aerobic exercise program, including intensive exercise, that the incidence and age of dementia are improved. So people who exercise have less dementia, and when they do get it, it's at a later age. And when they do get it, it's milder initially. And so whatever it is about exercise, chemokines, stem cells in the bloodstream. Now, much of that is not controlled data. So maybe people who exercise regularly also eat better and take better care of themselves. So it's complicated, but everything I know about what exercise does to the system suggests that the system favorably improves brain function as well. But I don't have a deep knowledge of the research literature on that question. Start exercising and eating well while you're young.
I tell students in class constantly, and especially about the cardiovascular health implicating all of your systems. In terms of your telehealth, I was wondering how it does for the telehealth modules, essentially, do they increase in difficulty for the patients, and how does that match up with what they're doing in the clinic?
Right, it's a really good question. In both clinic and in tele-rehab, the therapist modulates the degree of challenge. And so we built into all of our games that the easiest level has to be something anyone can do, and the hardest level has to be hard for us able-bodied people. Okay. And we have sort of 3 to 5 levels in between, depending on the game. And it's up to the therapist's judgment. The initial treatment assignment, we have an algorithm that takes the 33 questions from the Fugl-Meyer and suggests which games and exercises to start with, just to sort of standardize things. But then it's up to the therapist. If I had a bajillion zillion dollars, I would hire programmers to, what would you call it, a neural net, to look at what works and come up with suggestions as to when it's time to suggest what. But that's not a forte in the lab, and I also don't have a bajillion zillion dollars. But obviously that's something the field will be doing eventually, is understanding which patient measurements can inform which logically suggested alterations in therapy. Right now we rely on the judgment of expert licensed therapists to progress therapy in a personalized way.
Do you think compliance factors into like the increase in difficulty at all? Or like if you eventually got to the point where you introduced AI, like the actual modules themselves, is there variations like within different patient demographics in terms of their compliance with actually completing the modules?
Yeah, it's a really interesting and complicated area. Some patients, it gets into one aspect of motor learning, which is feedback. Mm-hmm. Some patients need to see that they're doing better and need to be told that, right? Some patients, it's just a matter of don't show me failure. The challenge level varies. Some people are slackers and some people are not. Some people, if you challenge them too much, you lose them. Some people, if you don't challenge them, right? And this is what the therapist is consciously or unconsciously calculating, which an AI system would pick up on, with the right kind of opportunity. The best I can say is people come in all different stripes and flavors with regards to the important issues you're describing. And it takes a good therapist to get a sense of that. And what we try to do is give them the tools in the telerehab system to be able to modulate those things. So they can adjust several difficulty levels in each game, and they can adjust the duration of the game. And we give a lot of feedback. We can get into that, but at the end of each game and at the end of each session, they get feedback as to how they're doing. When they're not progressing, that can come back and bite you. So it's complicated to know what to do.
Thank you. So are there any other questions from the audience? I know I've gone a little bit over time, but I keep asking follow-up questions. All right, if there's no other questions, I'd like to thank Dr. Kramer for an excellent keynote on behalf of myself as well as the organizing committee. As I'd also like to thank all of the other speakers today, as well as our student presenters. With that, I believe I'm turning things back over to Dr. Dietrich. Yes, perfect. Thank you all.
Okay, well, thank you, Courtney. Thank you very much. And again, Stephen, thank you very much for an outstanding talk, and all the other individuals that talked during this conference. I thought it was fantastic. This is our 5th year, and each year gets better and better. And we have a growing momentum at the University of Miami in terms of bringing engineering and biological approaches to target these very complicated problems. So it's an exciting time. So again, as Sarud mentioned this morning, we want to thank everyone for the organizing committee for doing a great job, the students for giving great presentations. Thank you, Erica Swazo, very much for being behind the curtain to actually help us put all this together. And Iggy did a fantastic job again. And next year, hopefully, this is our 2nd virtual meeting in a row. And hopefully next year, we'll be able to have a face-to-face meeting with all our colleagues throughout the state of Florida and elsewhere. And again, invite really outstanding individuals, thought leaders to present their work. Sarud, any final comments?
No, I think you summarized it very, very well, Dalton. Thank you, everybody.
Thank you, everybody. See you next year. Take care.